So hello dear student, welcome back. Welcome back to the GDC English channel. So in today's class, we are going to discuss 200 questions that are going to be really very important from your Tamil Nadu drug inspector exam point of view. Okay. So yes, these questions are being specially designed in a way that they will help you in your exam in a same way that today they are going to help you in order to recall all the topics, all the things that are you done for the preparation of your exam. Yeah. So without going late, let's start our today's discussion. So I am taking first 65 question of that particular session that is going to be from your two subjects. One is pharmaceutics and another is the pharmaceutical jurisprudence. The both topics are going to be very, very important from your exam point of view. Okay, so we taking both the important topics for that particular session. So yes, start our discussion. The first question of our discussion is mainly saying that who is the father of Indian pharmacy? Okay, give me a minute. Give me a minute. Give me a minute. Just give me a minute. I am just going things in the right way. There's something wrong with the PPT. Okay, I think now it is done. Yeah. Okay. So we are going to talk about the first question, or we are taking now the first question of our class. The first question is going to be what? They are mainly asking about what is the father of Indian pharmacy? What is the father of Indian pharmacy? The option A is the PC Ray, Praful Chandra Ray, ML Shroff is the second option, RN Chopra is the third option, and the last one is the TK Gajjar. Yes, answer this question fast. Whoever taking the class. Yeah, are you getting my voice? Okay. So let's start our discussion. Option B is correct. Yes. Option B is correct. Yes. ML Shroff, Mahadevi Lal Shroff is known to be as the pharmacy or the father of Indian pharmacy. Father of Indian pharmacy. What he done for the pharmacy? Firstly, he stabilized the BSc course at BHU. The second thing he done is the integrated course on BHU is also stabilized by same person, that is the ML Shroff sir. The third thing he has done is mainly starting of a journal that is known by the name of Indian Journal of Pharmacy. Yeah, Indian Journal of Pharmacy. That is later named to be as the Indian Journal of Pharmaceutical Sciences. The same name is being given to the same journal after some time. Okay, Pharmaceutical Sciences. Yeah, the PCA. The PCA is known to be as the father of Indian organic chemistry. Indian organic chemistry. Yeah. The RN Chopra is the person who is the main person behind what committee? The Chopra committee. The Chopra committee that is being formulated in the year of 1930. That is also known by the name of Drug Inquiry Committee. Okay. Can anyone tell me the date at which the Drug Inquiry Committee is being followed? And in a year of 1903 and 1907, a person who made two companies in Mumbai itself is mainly the Professor T.K. Gajjar. Is the person that is behind the establishment of the two companies. Firstly, in 1903, a small company that and a large company that is known by the name of Alembic Pharmaceuticals in a year of 1907. So yes, the answer for that particular question is going to be B, absolutely correct. But the other information that are related to the given questions are as follows. Okay. So moving towards our next question, the pharmacopoeia is being published by. The pharmacopoeia is being published by. Yeah. If you want me, if you want to recall me certain aspect or the points that are being given by the Chopra committee, then you can also do it in a comment section. Yeah. So the moving towards our next question, the pharmacopoeia is being published by the Ministry of Education is publishing it, the Ministry of Health and Family Welfare is doing it, the Ministry of Finance or the Ministry of Commerce. What is the answer? So I can give you a hint. The ministry that was earlier, that was not earlier, that was now the funding ministry for the NIPER itself. Funding ministry for the NIPER itself. Yes. So answer is going to be the Ministry of Health, Family and Welfare. So and you have to put in between them. Okay. Earlier the NIPER is being funded by the Ministry of Chemical and Fertilizer, but nowadays it is being published by or it is being funded by the Ministry of Health, Family and Welfare. Okay. The pharmacopoeia is being published time to time, and it is one of the point that is being given by the Chopra committee. Okay. And under the guidance or under the leadership of R.N. Chopra sir, the first pharmacopoeia list is being prepared. Okay. Coming towards our next question, the B is the correct option of it. Which of the following is a factor influencing the dose calculation? So what is the different factor that are being listed? So either the synergistic effect, it is the synergistic effect is further divided into two effects. The first is one is mainly saying to be the additive effect. Yeah. What is this going on? The second effect, it can be the supra-additive effect. We have multiple or plenty of options that are mainly showing the additive and the supra-additive effect. The another one is also going to be the antagonism effect. So we have to just increase the dose if some impeding factor or some antagonizing factor are being present in the case. But if certain facilitating factor are being present, then we have to just decrease the dose. So yes, in case of supra-addition or addition that is a part of synergistic effect of a drug, we have to just decrease the dose of that particular drug. But in a case of antagonism, means we can say that it is a kind of impeding factor that is resulting in the decrease in therapeutic activity or ability of a drug. So we have to increase the dose of our given drug whose effect we want in our body. The idiosyncrasy is what? The idiosyncrasy is mainly the genetically predetermined reaction, genetically predetermined effect that will lead to the drug allergy. Okay, at what dose? At what dose? So there are genetic effect or genetic makeup that will mainly lead to the allergy to a certain drug is said to be the idiosyncrasy. In the same way, if we say about the microbes, then the development is known to be as the resistance. But if we are mainly saying about the humans, then it is said to be what? The tolerance. And the process of rapid development of tolerance is known to be as what? Rapid development of tolerance is known to be as the tachyphylaxis. Yes. So yes, all of these factors are going to affect our dose calculation. In some factor, we have to just decrease the concentration of our drug. In others, we have to just increase the factor dose of our drug. So D is going to be the correct option. Moving towards our next question, the dose of a 300 mg is for an adult. What is the dose of appropriate for the 7-year-old who's having its weight 48 lbs? So yes, firstly, you have to know that lbs, 1 lbs is equals to how many kgs? Around we can say 554 kg. That is approximately weight a half of kg is the value in kgs related to the 1 lbs of weight. So what you have to do is that is only the calculation. They are just asking about the dose. The dose that is being appropriate to be given. The dose is 300 mg. Is for an adult. What is the dose? The adult dose is being given. The appropriate dose for the 7-year-old child. 7-year-old child. The weight of the child is being given here. Okay. So adult dose is being given. That is the 300 mg. Adult dose we can. Yes, give me the answer. Each and every question I explained you. Sir, please increase volume. Okay. Let me hear once. Yes, give me the answer. Each and every question I explained you. Give me a minute. Solve this question. And what formula you are going to apply here? Yes. Also tell me that particular question. So now you are getting my voice. Now you are getting my voice or not? I think now I am audible to you guys. Yeah. Are you? And what formula you are going to apply here? Yes. Also tell me that particular question. So now you are getting. Okay. Now it is okay. Now you are okay with my voice. I think you all are okay. Yes. Yes. What is going to be the answer? So what formula you have been applied here? Formula. So this question is mainly from the clinical pharmacy. Clinical pharmacy or dose calculation. Okay. The answer for that particular question is going to be 96 mg. Okay. Coming towards our next question, identify the reason which may be responsible for increase in the size of dispersed particle in the suspension after the aging. So during the aging process, what is the main effect that will lead to the aggregation of particle or increase in their size? On the basis of that particular property, we can say it is a kind of, they are talking about the instability of suspensions. Yes, instability of suspension. Either it is a Ostwald ripening, either it is mainly the temperature cycling, that can also be said as the freeze thaw freeze cycle. So freezing is mainly decrease in the temperature. Then we have to put the particular sample to the certain temperature. Then we are again freezing it. So this is known as a freeze temperature cycling or the freeze thaw freeze cycling. The polymeric transformation can be there. The Ostwald ripening. What is going to be the correct answer? What is going to be the correct answer? Yes. So I think so. Over the period of time, we are two kind of suspension. We are mainly having the two kind of suspension. That can be the flocculated type of suspension. The another one is mainly the non-flocculated or deflocculated type of suspension. So what is the pharmaceutically accepted type of suspension is the flocculated type of suspension, because in the flocculated type of suspension, the aggregate will be formed that can easily be redispersed. Okay. By certain shaking, we can easily be able to redisperse them. Yeah. So yes, over the period of time, the Ostwald ripening is affecting the size of the dispersed particle, because only two things we are mainly having in our suspension. The one, that is the major part, that is being known by the name of dispersion media, and the small particle that is distributed throughout this media is known to be as what? Are known to be as the particle of dispersed phase. So the stability or the overall stability is being not only related to the particle property of the dispersed phase, but is also being related to the viscosity and some other factors related to the dispersion medium itself. So yes, Ostwald ripening is going to be one of the factor that is going to responsible for it. Freeze thaw freeze cycle is also a factor, because sometimes the temperature will also induce the nucleation or aggregation. Nucleation is a factor that is being result into the formation of crystals, specifically if we say. The polymeric transformation, we can say yes, the polymeric transformation, because all the polymer will only differ in their chemical property, but are mainly same in the property of like chemical property is same for all the polymers. Only the physical properties like the melting point and etc. is mainly different for all the polymers. So yes, the polymorphic transition or the transformation can also be a factor. Okay, here or incompatible polymer addition can also be a factor here. Polymorphic addition can also be a factor here. Okay, not the polymer. They are mainly talking about the polymer. So D is going to be the correct answer. Moving towards our next question. The crystal habit of an drug is important for suspension due to. So why the crystal habit? So crystal habit is what? Crystal habit is nothing. Crystal habit is just the external appearance of external appearance. So if a single crystal, single crystal can have more than one crystal habits. In the same way, a single person can wear different kind of clothes. Okay, in a day, he or she can wear around 2 to 3 types of cloth or a single type of cloth. So in the same way, the crystal or the amorphous, crystalline and the amorphous property are mainly related to the bulk. They are the mainly bulk property. But the crystal habit is only the external appearance. So we can say like a single crystal can have a different crystal habits or single crystal. So the crystal habit of a drug is important for the suspension due to its redispersibility, sedimentation, and physical stability. Why the crystal habit is going to be important? Why the crystal habit is going to be important? So we can say the crystal habit is a kind of external property or not a bulk property. So yes, the crystal habit is certainly related to the redispersibility or the aggregation, we can say. So if the aggregates will be formed, the overall gravitational force that is being acting over a given particle in a suspension will also going to be increased. So if we consider that this small particle will mainly feel a lesser amount of gravitational force as compared to an aggregate that is being formed. So the ability to settle down is more in that particular larger particle or the aggregates, we can say. So yes, the sedimentation is also being effective and the physical stability is also being effective. All the following things are being affected by the crystal habits. Okay, in case of suspension. So D is going to be the correct answer. Okay. So yes, the seventh question is the pre-formulation study involves. What are the pre-formulation study involves? So we can, in an easy language, we can say like physiological or the pre-formulation is being done in order to minimize the time, in order to utilize all the things, either the class 1 of excipient, class 2 of excipient, I think you have read about them, or class 3 of excipient in a better way, while knowing all the physiochemical properties that are being related to our drug as well as with our excipients. So yes, the pre-formulation study is nothing. It is just the study of physiochemical properties of our drug. That is a bifurcation of two words, the physical property first, and the second is mainly the chemical property. The chemical property that is being related to the chemical structure of the drug, and the physical property like the melting point, etc., that is mainly related to a physical appearance of the drug. Okay. So yes, on the basis of the chemical property, I can give you an example like hygroscopicity that can also be related to the chemical characteristic of a drug. Okay. So yes, the physical pre-formulation is development of a suitable dosage form for a drug. Determination of the pharmacological action. Is it related to the pharmacological action? No, this is not related to the pharmacological action. Determining the molecular structure. No, we are not doing any kind of molecular modeling. So we are not consider the molecular structure at all. And the adverse drug reaction, the ADR, is also being studied in the toxicology. That is a kind of a branch that is associated with the pharmacology itself. Yeah. So the only thing we are mainly doing, developing a suitable dosage form from a drug or for a drug. So yes, the API that is initially known to be as the drug substance is being converted after the addition of suitable excipient, is being converted to a formulation that can be known by the name of or said to be the drug product. Yeah, the formulation can also known by the name of drug product. Most of the students are get confused there. So yes, A is the correct option. So yeah, the match the following question: the true density, granular bulk, and the bulkiness. What are going to be correct option for them? So the two density, which equipment is being utilized for the measurement of it? Yes. So we have to start with the last, the bulkiness. The bulkiness, we can say, is just the reciprocal of bulk density. We can write in the same way. So yes, what option is correct? So we can say fourth is mainly for the B. Fourth is mainly for the P. Yeah. Fourth option is mainly for the P. Okay. The true density is mainly studied by utilizing the noble gas. One of the noble gases mainly the helium pycnometer. Helium pycnometer. Yeah. The granular density and the bulk density is being determined by. So if we are talking about the bulk, we are mainly in a gross talking about all the powder mass, So yes, for that purpose we can also utilize. So this process is not that much precise. We are mainly using the graduated cylinder, but the granular density, for the determination of granular density specifically, we have to just use the mercury displacement method, because mercury is a thing that will not go inside the intraparticle space. Okay, it will mainly cover in this way. Yeah, so I think so. 1 4 R and 4 P. So third option is going to C option is going to be correct. Moving towards our next question, the Venturi meter is suitable for which type of installation? The batch installation, the temporary setups, the online installation, and the laboratory setups. Yes, for what the Venturi meter is being suitable for? Where is your chat gone? Yes, answer this question. The Venturi meter is stable for which type of installation? It is not used for the batch installation. It is not a temporary setup for which we are going to such a large equipment of Venturi meters. Okay. So the thing that online installation, for the online installation, we are going to use the Venturi meter. Okay. Not at the lab scale we are going to use. C is the correct option. How many of you are giving that answer? Yeah. Moving towards our next question, which one of the following pair is incorrectly matched? So you have to just find the incorrect pair out of the correct pairs. So yes, the lipophilic antioxidant that are also known by the name of blocking agent are what? Are BHA, benzyl hydroxyanisole, benzyl hydroxytoluene, and what? The hydrophilic antioxidant that are mainly the ionic form or the sodium salt itself. Yeah, that is the sodium metasulfite, sodium metabisulfite, sodium sulfite are the some of the hydrophilic antioxidants. Yes, we can say these two options are correct. Yes, the citrate buffer we are going to use. What is the pH range for that citrate buffer? pH range for citrate buffer. Tell me. The carbopole, the carbopole is being used as a preservative. No, no, this is not correct. So where this carbopole is being used as an, is it a diluent for the tablet? Is it an anti-adherent? Is it what? Yes, answer this question as well. I'm waiting for your answer. Indian Pharmacopoeia Lab is in Ghaziabad. Yes, National Institute of Biologicals is also in Noida. Okay, so what are being prepared in National Institute of Biologicals that are located in Noida? Yeah, somewhere remember that surface antigen for hepatitis A, hepatitis C. HIV determination kit are being manufactured here. Just give me a minute. Okay, so C is the correct option. We have to just find the incorrect one. Moving towards our next question, the elasticity, not the elasticity. The elasticity of a soft gelatin capsule shell can be controlled by adding the elasticity of a cell of a soft gelatin capsule is being determined by. बताओ. What is the reason behind you are answering that particular option? Yes. Yes, 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 yes. The elasticity is mainly related to what? The elastic property of any formulation is being related to the plasticizer content into it. Yes. So if we vary the plasticizer content in any formulation, any form, can you tell me other thing for hard gelatin capsule and soft gelatin capsule? What are the bloom strength for them? What are the moisture content? Yeah, 6 to 9%, and this 13 to 16, I think so. The bloom strength is high for the hard gelatin capsule and soft gelatin capsule. The only single shape is being present in the case of hard gelatin capsule, but multiple shape can be for the soft gelatin capsule. Yeah. The pharma gel or the gelatin A form, yes, is mainly responsible for the formation of that particular. So yes, we are talking about the plasticizer. The plasticizer is mainly related to the elasticity, and the most commonly used plasticizer in any formulation whenever you're studying about in your pharmaceutical technology subject, the most commonly used is mainly the sorbitol. The sorbitol, and more because sorbitol is mainly belonging to the class of, if they are asking about the class, they are either giving you the option polyols. Yeah, polyols, and the best member out of these polyols are going to be the sorbitol. Okay, coming towards our next question. The Creep test is being analyzed for the viscoelastic property of. The Creep test is being elastic for the emulsion, for the lotion, for the ointment, or for the suspension. Answer this question first. The Creep test is for. The Creep test is for. So yes, whenever we are mainly relating or discussing about the formulation that is mainly forming a thin film over our skin, we have to just refer a single formulation. That is what? That is what the creep test is. A single formulation that is also contain the particle into it and having the two action, that is mainly the emollient action, and the second action is mainly the protective action that is associated with them. So yes, it is related to the ointment. Coming towards our next question, identify the propellant in the pharmaceutical aerosol is carried out by the identification of the protocol of the propellant, that is also known by the name of the heart of aerosol. The heart of aerosol. So it is mainly done by the gas chromatography, tag open cup type of apparatus, the pygnometer, and the IR radiation. So we can remember it with the trick IG of police. So P is mainly stand for the propellant, I is mainly stand for the IR spectroscopy, and G is mainly stand for GC, that is the gas chromatography. So yes, the gas chromatography and the IR is going to be the answer. P and C is correct. P and S is correct. So option B is the answer. Option B is the answer. Okay. 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 So yes, pygnometer, they are mainly utilized for the density. The pygnometer and the hydrometer are being utilized for the hydrometer. Okay, for the density determination in case of aerosol. Yeah. So yes, answer B is correct. Coming towards our next question, the water attack test is being used for the determination of. So there are certain four kind of glass, 1, 2, 3, and 4. All the glasses will mainly, so this is mainly the highly resistant glass, class 1. Class 2 is mainly what? Soda lime or treated soda lime? Treated soda lime glass. Okay, soda lime. The class 3 is mainly the soda lime itself, soda lime glass, and the class 4 is the multi-purpose glass that is not used for the aerosol but is being used for the tablet, for the capsule, etc. So all of these are going to give the powder attack test, except the second one, the type 2 will mainly give the water attack test. Powdered glass test is being given by all of them, and water attack test is mainly given by the second one. So what is the resistance of coating tablet for moisture? We are not discussing about the tablet. The resistance of empty gelatin capsule they are discussing about? No. No, the chemical resistance of a glass. Yes, they are mainly related to the glass. Yes, this is the easiest question can ever exist. So C is the answer. In the fourth option, they are discussing about the surgical dressing. No, this is not related at all with the surgical dressing. So yes, C is the correct option of it. Yeah. Going out toward our 15th question, which of the following is an example of thermosetting plastic? So the plastic that will mainly form a hard mass whenever we are heating it up. That is known by the name of thermosetting plastics. Yes, answer this question. What is going to be the thermosetting plastic? Not the PVC, not the PVC, not the polyester, not the polypropylene, not the polyethylene. I am writing here polyethylene is not that. Yeah. So if we say certain kind of PVC, where PVC is going to be used, you have also remember where polyethylene you are going to use. Yeah. For the IV catharsis, we are going to use the PVC. Polyethylene. Polyethylene is being used for the eye drop storage or eye formulation storage. So we are answering, we are mainly the question related to the thermosetting plastic. So thermosetting plastic is the only one that is mainly the urethane. Urethane. Okay. Coming towards our next question. Which theory stated that the energy required is proportional to the ratio of size change? Either it will be the Rittinger's theory, Bond's theory, Cakes theory, or attrition theory. What is that particular theory is? Kabir, you can remember with that particular trick. That is the Cakes theory. B is mainly stand for the Bond's theory. Either R is mainly stand for the Rittinger's theory. All these theory is mainly related to the size reduction or the diminution we can say, or we can say the communication. Yes, communication भी बोल सकते हैं उसको. So in the same way, these are related to the reduction in the particle size. So they are specifically asking about the energy relation with the size reduction. E is directly proportional to the size change is mainly shown by is mainly given by what? So when we kick a given stone, that it will mainly the size of that particular stone is being reduced if we kick the given stone with a certain higher amount of energy. So yes, whenever they are talking about the proportionality in between the energy and the size reduction, they are talking about the Kick's theory. Yeah. So C is the correct option. Coming towards our next question, the lauryl amino propionic acid in the shampoo is used as the lauryl amino acids in the shampoo is being used as a detergent. No, they are either asking about the in the detergent they are going to use either the anionic detergent, either the cationic detergent. But whenever they are asking about the baby shampoos, they are mainly utilizing nowadays cornstarch is being used for that purpose. The pH adjusting agents, the conditioning effect, the clarifying agent. Clarifying agent, it is not the clarifying agent. The lauryl aminopropionic acid, LAP. LAP is what? It's certainly the conditioning agent. Can you tell me the other name or other conditioning agent that are being used for the conditioning purpose in the shampoo? Yeah. So B is the correct option of it. Coming towards our next question, because we are time bound, or we have to just solve you the 200 questions. That's why the thioglycolic acid like compound have application in the following type of cosmetic formulation. So what they are asking about the thioglycolic acid components. Yes, I can. I taught that particular subject multiple time. I taught that particular thing multiple time whenever I was taking your classes related to pharmaceutics. Yeah, specifically the topic that is the cosmetics. The disulfide bond, the disulfide bond that is present in hair, somewhat creatinase enzyme is also being worked in that particular formulation is being included in that particular formulation. So what formulation are going to be these? Is they epilatory or depilatory? So yes, if they are mainly doing the complete exit of the hair from our body, then they are known to be as the epilatory preparation. But here they are not containing the creatinase enzyme. They are only asking about the 𝘂𝙀𝙀𝑉𝑖c acid. So yes, they are the depilatory preparation. Coming towards our next question, which one of the following is not a major pharmacokinetic method for bioavailability assessment? So they are specifically asking about not a method. All the others are the method, but this is not going to be a method. The plasma level time study, the urinary excretion study, or the urinary excretion rate, we can say, the pharmacodynamic response study. So we are discussing about what? The bioavailability. The bioavailability. Yeah. And none of the above. Yes. Waiting for your answer, guys. Participate in it. What is the answer? Yes, C is the answer. The plasma time graph is being used for the study of bioavailability. The urinary excretion rate, or the urinary excretion rate, we can also say, is also utilized for that purpose, but not majorly the pharmacodynamics. That is mainly related to what? That is mainly related what drug does to our body. And the bioavailability is a term that is mainly related to what? That is mainly related to the pharmacokinetics. That is what our body does to the drug. So yes, this is not a major part. So C is going to be the correct option. Coming towards our next question, consider the following statement regarding the statistical movement theory. The statistical movement theory can be used to determine the relationship in between the mean dissolution time in vitro or mean residence time in vivo. In vivo is the condition whenever we are experimenting or doing experiment in our body, human body. In vitro is the condition that is being set inside the lab. Okay. The second point is the mean dissolution time in an in vitro parameter is an in vitro parameter, or mean residence time is an in vivo parameter. What is going to be correct? And the last one is the statistical movement theory cannot be applied to dissolution studies. They are asking about the statistical movement theory. So what are the points that are going to be correct? That are going to be correct. So statistical movement theory cannot be applied. No, it can be applied for the dissolution study. So whenever they are asking about the lab setup, so yes, we are talking about the MDT, that is the mean dissolution time. But whenever we are talking about the things that are going inside our body, we are mainly saying about, or we are mainly taking into consideration the MRT, that is the mean residence time, because it is the time to which a drug is remain inside our body. The statistical theory can be determined the relationship in between the MDT, yes, the MDT and the MRT in vivo. So yes, they are mainly useful in determination of IV, IVC correlation, in vitro, in vivo correlation, that are having their different class, the grade A, the grade 2, the grade 3, on the basis of their evolution, on the basis of the correlation. All these things is being stabilized. So yes, the fourth option is not correct. A is the correct option of it. Coming towards our next question or discussion, the patient receives 500 mg of a drug IV routes. So yes, after distribution equilibrium, the plasma drug concentration is 10 mg per liter. Calculate the apparent volume of distribution. They are just asking about the volume of distribution. There are different formula for the volume of distribution. We can say X by C. X by C is the common formula for the volume of distribution, but they are asking about in relation to the loading dose, because they are mainly giving it. So the loading dose can be written as the loading dose is the factor that is only affecting the loading dose is going to be the volume of distribution into the desired plasma concentration. The desired plasma concentration. So yes, we have to just calculate the volume of distribution. The loading dose is given here, that is the 500 mg. And what is the desired plasma concentration? We have just to divide that particular thing, and then we will get our answer. So solve this question. What is the answer? I have already solved it for you. Yes. Why are you guys taking that much time? This is a simple formula. You have just to divide the loading dose with that of your desired plasma concentration. Why are you guys taking that much time? Answer this question fast. Not even a single comment is coming from your side. in relation to that question. Earlier, you are responding for a single line of question, but as you know, in drug inspector exam point of view, the numerical question are also going to be important. Okay. So what is the answer for that particular question? Yes, the answer is going to be, I already have been solved that particular question. Yeah. Still, you are not giving the answer. Still, you guys are not giving the answer. Whenever you divide 500 with 10, the answer will be always, always what? 50 liter. The unit for perfusion can animal 10 be? The unit for perfusion. The unit for perfusion, you can say ml, minute ml is going to be the unit for perfusion. Okay, coming towards our next question. Yes, this question is mainly related to the dose response curve, DRC, I think so. So the consider the following statement regarding the area under the curve. So AUC represent the total integrated area under the plasma concentration time profile. AUC express the total amount of drug that will reaches to the systemic circulation after administration. AUC expressed in the microgram per ml into the R. AUC is the most important parameter for evaluation of the bioavailability of the dosage form. So yes, all these things are, all these lines are mainly saying the same things. So whenever you are discussing it with the teacher who teaching you pharmacology, the only thing that will mainly determine the bioavailability is the area under curve. So yes, the bioavailability can also be written to be as what? The AUC for the test dose divided by the AUC IV, because IV will having the 100% of AUC. That in turn related to the bioavailability. Yes, this is correct. The AUC expressed in microgram per ml per hour. Yes, will this is also going to be correct. The total amount of drug that will reach the systemic circulation after administration. Yes, this is an also a definition of bioavailability itself. Okay. If they mention in unchanged form. Okay. So yes, area and curve are the total integration under the plasma concentration time profile. Total integrated area. Yes, this is a total integrated area. If this is a bell-shaped curve, so this is mainly the total integrated area. Okay. The Cmax lie here, somewhere here. The Tmax somewhere lie here. If T is it, C is it. Yeah. So all the statement are going to be correct. Moving towards our next question. A drug has an apparent volume of distribution. The VD is mainly given. The volume of VD is mainly given 80 liters. Okay. If the 400 mg of drug is present in our body, then the plasma drug concentration is. Then the plasma drug concentration is what? XYZ. Yes, answer this question. I'm giving you 2 minutes in order to solve these kind of simple questions. But you have to answer this question by yourself. I'm not going to solve it for you. Yes, tell me the answer. Tell me the answer. Okay, moving toward our next question. Okay, so yes, this is what? This is what we have to just divide the volume of distribution to what? The drug present in our body to the plasma concentration of the body, because the volume of distribution having its formula, the drug or the total drug inside body divided by what? Divided by the plasma concentration of it. So they are just asking about the plasma concentration. We have to put here volume of distribution. We are moving here. So this is mainly where 400 divided by 80. The answer comes to be 5 mg per liter. Okay. Moving towards our next question. The CGMP subpart G is related to the subpart G of CGMP is related to packaging and labeling control, laboratory control, records and report, holding and distribution. What is this question? The subpart G is related to CGMP. Is it not the cyclic GMP? Because it is written in the same way as the cyclic GMP is being written. Yes, I think so. You just have to remove that particular C from here. The G, where the G, that is the good manufacturing practices, that is comes under the schedule M, that will having its different part. The part 1 of it, the part 2 of it, that is related to the area that is being needed for the production. So in case of uncoated tablet, how much area is being needed as a primary area or the auxiliary area, you can also say. So in the case of uncoated tablets, we have to write here 60 meter square and 20 meter square ancillary area is being required. So in the same way that is being given for the different purpose, and you also remember with a trick like schedule M1, M2, and M3, that is HCM related the manufacturing to homeopathic, cosmetic, and manufacturing of medical devices. Yeah. So CGMP, the G is mainly related to the packaging or labeling, the laboratory control, the record keeping. No, you have to keep the record. Schedule U is mainly related to the record keeping. Okay. The laboratory practice is being related to the schedule L1, that is the replacement of schedule what? That is mainly come in a year of 2010. The same year in which the friability testing for the tablet is converted to or being transferred from the unofficial test of the tablet evaluation to the official test for the tablet evaluation in the same year. Okay. So yes, it is mainly related to the manufacturing, packaging, labeling are all related to the good manufacturing practices. Okay. And what kind of thing is related like spurious, misbranding, or adulteration is related to the packaging and labeling? You have to answer that particular question as well. So yes, the good laboratory practice that is come under the principle is mainly based on the ICMR guideline, OECD guidelines, WHO guidelines, Schedule Y. Schedule Y is related to somewhere the import and the manufacturing condition that are being needed for the import and manufacturing, and also the things that are needed for clinical trial conduction. Okay, phase 1, phase 2, phase 3, and phase 4. So yes, they are asking about the GLP comes under the schedule G. L1 introduced in a year of 2010. What are the guidelines behind it? The guideline that are being behind it, not the ICMR guideline. ICMR is not related to provide any kind of guideline. It is mainly a council of medical research. Okay, WHO guideline, this is not. Schedule Y, I have already explained you. OECD are the guidelines that are being related to the principle of GLP, good laboratory practices, we can say. The material awaiting the quality control approval should be stored in. So they are mainly asking about the awaiting material for the quality control, QC. Okay, is the dispatched area, is the quarantine area, is the finished good area, is the rejected material. It is not be the rejected material, it is not be the dispatched material, because only after the qualification of the quality control test the given material will be dispatched. It is not the finished good area, it is being as the awaiting is being done. So yes, we can relate from here the quarantine area where the particular formulation is being waiting in order to get the quality control approval. Okay, so B is the correct option. Coming towards our next question, which one of the following is not correctly matched as per of the classification of clean room under ISO 14644? So they are asking about is not correctly match with the ISO. So yes, the part 1 of ISO is related to what? The classification of air cleanliness. For what we are mainly utilizing the HEPA filter, the ALPA filter, that will mainly not allow the size of particle greater than 0.3 micrometer, and the airflow from them is 100 plus minus 20 feet per minute. Yeah, and all the other things. The 99.97 thing, you are also remember the 99.99 things particle or the air particle remover, you also remember. The test method is in the 3. Yes, the test method in the 3. The design, the construction, and the startup is also included in the 4. But the term and definitions are not included in the 5. So in what part the terms and the definition is being included? Yes, answer this question as well. So D is the correct option for that particular question. And I have also asked you a question, answer that particular question as well. Okay. So which department is primarily responsible for the initiation of first phase of OOS investigation? The production, the human resources, the quality control, or the sales? So human resources is not related to at all. The production department is only related to the production. The sales is related to the sale itself, but the quality control department is related to the primarily responsible for the first phase of the investigation. Okay. So yes, the quality control is being done over the formulation, then it will be dispatched in the until it is being stored in the quarantine area until the quality control report is not coming to the favor. Okay. See the correct option. The non-ionic surfactant based multilamellar, unilamellar, vesicular structure are called. So they are mainly asking about the multilamellar or unilamellar, specifically the non-ionic surfactant based. As the name indicating, you can easily be able to answer. The microsphere, the liposome, the neosome, the nanoparticle. What is the answer? What is the answer as the name indicating? So yes, we have to take N here or ION from here. So this is mainly become NIO. That is mainly indicating whenever they are saying that the formulation is a non-ionic surfactant, we are saying that it is neosome. Neosome. Yeah. So yes, C is the correct option. Very easy question. 𝙂𝙋𝘼𝙎𝙬. 𝙂𝙂𝙎𝙬 is being a sustained delivery system, is a kind of what? For the pilocarpine, for the topiramide, for the phenylephrine, for the latanoprost. Yes. 𝙂𝙂𝙎𝙬. They are asking about the 𝙂𝙂𝙎𝙬. The most commonly used drug that is being used for the purpose of glaucoma and all the eye related things that are belonging to the class of your what parasympathomimetic class of drug, that is going to be the pilocarpine. I is mainly indicating that it will having the 𝘂𝙀𝙎 ring as well into it. So this is the answer from the medicinal chemistry point of view. The pilocarpine will mainly contain the 𝘂𝙀𝙎 ring into it. Yeah. Whereas some other drugs that are also used in the case of glaucoma are certain the beta agonist. Yeah. Beta blockers, that can be the beta blockers. The alpha agonist and the carbonic anhydrase are also being used for that particular purpose. The rivastigmine, the galantamine, the donepezil are the certain other kind of drug. So yes, the P is the answer. The C is going to be the answer. What is the correct sequence for the citric acid cycle? What is going to be the correct sequence for the citric acid cycle they are asking about? Citric acid cycle is also known by the name of TCA cycle. That is the tricarboxylic acid cycle. Tricarboxylic acid cycle. So yes, tell me the answer. Tell me the answer. Yes. So most of the reaction that are conducting in our body, outside our body, is mainly starting from acetyl coenzyme. So yes, we have to just put the acetyl coenzyme at the first position. Yeah. Secondly, it will mainly goes to someone citrus, that is the citrate. Okay, that is the citrate. Then after the citrate, after the loss of water and all, yeah, that it will mainly form the oxalosuccinate, the oxalosuccinate. Then, then, then it will mainly going to form what? Then it will mainly going to form fumarate at the fourth place, and lastly, by accumulation or by production of FAD, one FAD will get converted to one FADH2, and at last they are mainly going to form the OAA, that is the oxaloacetate or oxaloacetate or oxaloacetic acid, we can say. So yes, the R is mainly the first. The R is mainly the first. The single option can help you to clear the concept. So what is the typical shelf life of a whole human blood cell collected using the standard acid citrate dextran ACD or the citrate phosphate dextrose solution? So they are the certain solutions that are acting as a buffer. So what is the going to the period for them? What is the going to be the shelf life for the whole blood? Shelf life for the whole human blood. Answer this question. If utilized while utilizing these things. Yes. So this is a kind of information based question. This is going to be 21 to 28 days. Going to be the shelf life. Which one of the following does not fall under the category of primary source of marketing research? They are mainly asking about the primary source. Whenever any company wants to do the marketing research, it is a salesman, it is a dealer, it is a consumer, it is a trade press. What is it? They are asking about the primary source for marketing research. All the information that is being related to our trade is being given in the trade press. So yes, it is acting as a marketing source because some or less these all sources are somewhere inefficient to give you all the information related to the things that are going into the market. The only thing or the liable source from which you are get the primary source, that in turn the liable source from which you are mainly get the marketing research information is the trade presses. Yeah. Coming to our next question, all of the following are aim for the sale promotion except what is not the aim for the sale promotion? The inducing customer to purchase more item, to attract the new customer, to increase the taxes on the product, okay, to popularize the brand. So why they are have to increase whenever they are asking about the sale promotion? So why they are including the increase in the taxes on the product? No, this is not included. So C is the option. That is the easy one. You can easily be able to odd one out or figure out the answer for that question. Okay, so C is the answer. OTC is mainly stand for over the counter, on the clock. What is it? What is it? What is it? The OTC is mainly stand for. OTC drugs are specifically the simple kind of drugs. Yeah, that is being like in recent days, the syrup is being removed from the OTC list. Other drugs that are coming in that particular other categories are the drug belonging to the Schedule X, Schedule CC1. Yeah, other than Schedule CC1, that can also the Schedule G related to the prescription only drug. The Schedule H drugs are the other drug. The OTC drug are the drug that can easily be sold or distributed without any kind of license. So OTC drug are the over-the-counter drug. Okay, one of the following is not correct in the main objective of the Pharmacy Act 1948. So they are asking about not correct Pharmacy Act in relation to the Pharmacy Act 1948. Restoration of the pharmacy profession in its due place in the health services. Rising the status of pharmacy profession. Yes, it can be. Regulate the practice and profession of pharmacy. Yes. To have the common syllabus of pharmacy throughout the India. So yes, it is dealing with what? The common syllabus. Restoration of the pharmacy profession. So if you regulate the pharmacy profession, due place in, because if you are only allow a certain person that will having the certificate of pharmacist or a status is being improved by utilizing certain thing, like they are also recommended that you have to do a dispensing experience of 250 hours, not less than 250 hours, or the practical experiment not less than 500 hours they are mentioning, or they are imposed in that particular pharmacy act. So the common syllabus is not related to them, but the approval of the syllabus, the approval for the examination is being done by the pharmacy or under the pharmacy act. Okay, so this is not correct. Okay, the number of person elected are elected by the Pharmacy Council of India for teaching profession from the teaching profession. So yes, I think you remember that particular trick 611, 621, 111. These are the number of elected members. These are the number of what? Nominated members, or these are the number of ex officio. to automatically become the member of the pharmacy council. So yes, how many are out of them is mainly belonging to the teaching profession? Yes, yes, I am waiting for your answer. So yes, the 6 is mainly from the teaching. The 1 is mainly from the MCI. Okay. Here the 2 out of 1 is mainly from the UGC. 1 is mainly from the AICTE. Okay. And out of these 3, the 1 is the DGHS, DCGI, another one, and the director of what? And the director of Central Drug Laboratory are the ex-officio members. So yes, answer is going to be what? 6. The tenure of the nominated member on the Central Pharmacy Council is for the year of how many year the Central Pharmacy Council is being worked for? What is the tenure? What is going to be the tenure? Yeah. What is going to be the tenure? So whenever anyone asks you DTAV, 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 that was having its tenure of 3 years. Okay. Okay. And is formulated under under what section? Yes. 6 or 7 or 5? Under 5. The constitution and composition. Or DCC is under section 7, and CDL is under Central Drug Laboratory is under section 6. Okay. So these are the certain sections. There are different sections that are also related to what? That are also related to the definitions. Definitions are included in 3. Okay. Different kind of, like for drug inspector is being included in 3E. For the government analyst is included in 3C. So yes, we can say yes. Answer this question fast. We have discussed much thing about it. The tenure of the nominated members on the Central Pharmacy Council is for a period of normally of 5 years. Okay, coming towards our next question. Which of the following is authorized to compound, to prepare, to mix, to dispense any medicine on the prescription of a medicinal practitioner? So yes, there are 3 places. The drug store, that will having its definition under Rule 65, 15A. Okay, the druggist or the chemist, that are having its definition same but in a 15B. Okay, and the pharmacy shop where the pharmacist, and the only place where the prescription drug is being dispensed, is mainly the pharmacy. That is mainly comes under the Rule 65 of 15A. Yeah, I hope you guys remember all these rules and schedules. Okay, so yes, which can do the things: the D Pharm, the registered pharmacist, the B Pharm, a person having the 5 year in such experiences. The only thing that is needed here is the registered pharmacist. That's why you are mainly applying, whenever you are applying for the pharmacist examination, they have first asked for your pharmacist certificate, because only the dispensing can be done by the registered pharmacist itself. Okay. The 40th question of our discussion is the drug should be deemed to be as the misbranding. So where the branding is being mainly done? The branding is mainly done over the packing. So whenever the packing is mainly not done in a proper way, then we can say it is a misbranded drug. So yes, the labeling is not done in a prescribed way. Yes, this is related to the misbranding. The coloring, the coating, the polishing or damage is concealed or to make the drug to look more therapeutically effective as it is not actually. Okay. Made to appear better or the greater therapeutic value than it really do. Yes, this is also correct. So all the things are correct. All the things are. So yes, these are correct options. Okay. 41 question of our discussion is which schedule of the Drug and Cosmetic Act 1945 provides requirement for the operation of blood bank and preparation of the blood component? Yeah. Schedule I, Schedule S, Schedule R. Schedule R is mainly related to what? Schedule R is mainly related to the rubbers or mechanical contraceptives, we can say. Mechanical contraceptives that are going to be what? That are going to be what? The condoms in specific, that is being tested where? In CDTL, Chennai. CDTL Chennai, the same area where the BCG vaccine is also being formulated. BCG, Bacillus 𝘂𝙬𝙬 vaccine. Yeah, Schedule S is mainly for the standards related to cosmetics. Okay, and F specifically the this part of this rule is mainly responsible for that. F2 is related to what? F3 is related to what? Double F is related to what? Double F is related to the eye formulation. F3 is related to the umbilical tips, or F2 is mainly related to cervical tips. So F is mainly the part 12B. This is the X 12B of the schedule F is related to the conditions being required for the preparation of the blood component. Okay. Moving towards our next question, the Drug and Cosmetic Act rule therefore list the substance that should be sold on the retail only on the prescription of the registered medical practitioner. So yes, this is the same thing that is mainly given on the label as well in certain kind of drug. If you studied in your class, they are mainly asking about that the same line is being written over the label of what kind of drug. What kind of drug? It is the prescription only drug. Prescription only drug that are the schedule H drugs. Okay. So A is going to be the correct option. So V schedule V is mainly related to schedule V is mainly related to what? Yes, for the patent and proprietary drugs. Schedule X are related to the habit forming certain kind of narcotic drugs. Okay, so these are the other schedules. So schedule H is correct. This question came multiple times in Kerala DI 2017, Andhra Pradesh 2018, Rajasthan 2018. Same question is being repeating itself again and again. So the 𝘿𝙂𝙧 and insulin are dash drugs. So they are asking about which kind of drug the 𝘿𝙂𝙧 and the insulin is. These are not under the schedule H. Schedule B is related to the rate or the balance, we can say, or the cost of the testing that is being offered by your CDL central drug laboratories. Okay. The schedule O is related to the disinfectants itself, for which we are mainly doing what? The Riddle-Walker test. On the basis of the Riddle-Walker test, we are mainly defining three kind of disinfectants having 18 value, 10 and 5 values. Yes. So yes, the glibenclamide with G, you can remember with G, and the insulin are mainly the schedule G drug. Okay. Schedule G drug. Yes. Coming towards our next question. Analysis of the sample for the sera, for the serum, protein, vaccine, toxins, and antigen is carried out at the CRI Kasoli, the Posture Institute of Kannauр, the Central Drug Testing Laboratory, Thane. The Thane is mainly related to what? The intrauterine devices. Okay. The NIV is mainly related to what? The production of HIV kits and hepatitis B surface antigen production, hepatitis C surface antigen production. It is related to the Pasteur Institute is being Kannauр is being related to the production of OPV, that is the oral polio vaccine. The CRI Kasoli is the only one that is remaining in our question, that is remaining in our answer. So yes, the same thing if they are asking about the animal is being done where? Is being done in IVRI, that is located in Izzat Nagar, or we can also say Mukteshwar. Yeah. So coming towards the question, having its answer A. Coming towards our next question, how many representatives are nominated by the central government to the Drug Consultative Committee? So only two kinds of member are mainly present in DCC, that is formulated under section 7. Yeah, we have already discussed. A two member is mainly appointed by the central government. Only two kind of member is present in the Drug Consultative Committee, and one each pharmacist from each state. Okay. Yeah. So yes, they are asking about the nominated member that are nominated by the central government are having their number too. So yes, B is the correct option. Moving towards our next question. Which one of the following section represent the misbranded cosmetics under the manufacturing, sale and distribution of drug and cosmetics? Yes. Is it 15C? Is it 17D? Is it 17C or is it 14? Yes. This section is related to the manufacturing, sale, distribution of drugs under the Drug and Cosmetic Act. Give me the answer. Is it what? 17? 17C? Okay. Yes. Answer is 17C. The license issue for the wholesale for the drug other than those specified under the, they are asking about the other than. Okay. Specified under the C, C1 and X. So there is a chance of or case of retail, wholesale and vehicle. Yeah. They are asking about the first class, that is the other than CC1X. So the licensing issue is related to, the licensing issue is mainly related to 20A, 20B, 21A, 21B. So 21 is not related at all here. Only the thing lies in 20A or 20B. Yes, so whenever the license came in the retail shop, it is under the 20B. For the wholesale, it is also under the 20B. But for the vehicle, it is under the 20BB. Okay. Okay. So whenever they are asking about the application of it, the application for the retail is under 19. For the wholesale is also under 19, but for the vehicle is under the 19B. Okay. Coming towards our next question. The permitted synthetic, organic or natural organic color used in the cosmetic shall not contain not more than how much percentage? So there are two soldiers are going. In Hindi, we can say soldiers are Sainik. So the concentration that is being allowed for the Sainik is 2 ppm, arsenic is 2 ppm. Okay. I used to remember by that particular trick. So 4 arsenic, this is not going to correct. 1 ppm arsenic, this is also not going to be correct. 2 ppm arsenic, 2 ppm arsenic, bilkul. This is going to be correct. For the lead, yes, the 20 ppm is being allowed. Okay. The 25 ppm for lead, no. 100 ppm for other heavy metals other than lead, yes. 100 ppm for other than lead is being allowed. So yes, the C option is going to be correct. Okay. Moving towards our next question. As per of Drug and Cosmetic Act in 1945 and report of serious adverse event occurring during clinical trials to the subject after due analysis shall be forwarded to the licensing authority within how many days of its occurrence? How many days of its occurrence? So they are asking about during the clinical trials. Okay, if any adverse effect is being occurring, so you have to inform the licensing authority in how many days, a minimum interval of time for 10 days. Within 10 days, you have to inform the licensing authority if in a case the adverse event is being occurred whenever you are doing your clinical trials. Okay. So C is the option. According to a Drug and Cosmetic in 1940, whose rule come under 1945, if any person willing will obstruct an inspector for exercising the power, so the power of inspector is, yes, in which schedule? The power of or they are mainly asking about the drug inspector. Okay, the power confound to him upon him, he shall be punishable with an imprisonment of that may be extended to 3 years, 7 years, 5 years, or 2 years. Yes. for a period of not less than, yes, try to answer this question, for a period of not less than 3, not less than 3 years. Okay. Give me a minute. Not being able to see your comments. Okay. So 3 years is the answer. Coming towards our next question. The no color is permitted to be used in the drug except mentioned in the Drug and Cosmetic Act 1945 under. So no color is being permitted to be used in the drug except that are mentioned in which of the following rule under the Drug and Cosmetic Act? Yes, under the Drug and Cosmetic Act. So this is mainly related to not 126, not 128, not 129. This is related to specifically the rule 129, 127. That is mainly saying the list of drugs or the list of colors that is being permitted to be used in the color. Okay, so B is the correct option. The illicit trafficking is related to the narcotic and psychotropic substance, means cultivating the cocoa plant and gathering any portion of it, cultivation opium and any other cannabis plant, selling of metoprolol tartrate, and importing the 𝘿𝙂𝙎𝙎𝙎. So yes, selling of metoprolol tartrate is not the illicit trafficking. The 𝘿𝙂𝙎𝙎 you can easily be able to take from any pharmacist shop. The cultivation of cocoa and gathering any of its part is an offense. With the illicit trafficking is come under the illicit trafficking, or cultivation of opium and cannabis is also a kind of illicit trafficking. So A and B both are correct. Coming towards our next question, which of the following is not a psychotropic substance under the Narcotic and Psychotropic NDPS Act? These are the barbitals, these are the estazolam, albendazole, or mazindol. So what is it? Out of them, the only drug you find here is the albendazole. That is somewhere antiprotozoal. Antiprotozoal, I can say, belong to the class. So this is not come under the NDPS Act. Okay, so C is the correct option. The cultivation of the opium poppy and production of the opium are to be supervised by the narcotic inspector, the narcotic commissioner, the drug controller, the narcotic consultative committee. So how many members are present? How many members are present in the narcotic consultative committee? Yes, answer this question as well. So all the things are being done. Cultivation and the production of the opium is either being done in presence of the narcotic commissioner itself or any other person that is being empowered by the narcotic commissioner on his behalf. Okay, so C is the correct option. And the members for the narcotic consultative committee will having a 20 member, including the president itself. Okay. Coming towards our next question, as per of the NDPS Act 1985, what are the punishable offense for the contravention related to the coca plant and the coca leaf? Imprisonment for 5 years with 1 lakh rupees fine, 20 years with 2 lakh rupees fine, 12 years with 3 lakh rupees fine, or 10 years with 3 lakh rupees fine. So there are two kind of punishment is being done in the case of NDPS Act. For the first conviction, the punishment is mainly for the 1 lakh rupees of fine with 10 years of jail. For the second conviction, it will mainly become around 3 lakh rupees of fine with 20 years of jail, I think so. So yes, the residus imprisonment for that particular term. So the D option is going to be correct. Okay. Coming towards our next question, that is mainly related to talisman, mantra, 𝙂𝙀. That is, the act is being passed in a year of and come into force in a year of 1955, the same year in which the act, that is the Medical and Toilet Preparation Act, is also come into force in the same year. So this is mainly the Medical Remedies and Objectionable Advertisement Act, OAA we can say. Okay. Any other charm of any kind of which is allegedly possesses the miraculous power for or in the diagnosis, cure, mitigation, and treatment and prevention of any of the disease to the human and the animal, or for affecting or influencing any way the structure or the organic function of the human body, of the human being or the animal, is called the magic remedy, the medicine, The medicinal remedy or the tragic remedy? What is it? What is it? Yes, as it is related to the mag Magic Remedies Act or Objectionable Advertisement Act. So this is they are talking about the talisman, mantra and kavch. So they are related to the magic remedies itself. So B is the correct option. Under the Drug and Cosmetic Remedies Act, Objectionable Advertisement Act that we are earlier discussed, 1954, advertisement of certain drug for the treatment of certain disease or disorder that are mainly discussing about or telling about the Schedule J, the list of certain drugs, the list of certain name of disease and disorder and the condition, the list of certain drugs and the disease, and the type of advertisement. What is it related to? So as it is mainly related to the Schedule J of the drug, that are the list of certain drugs and the disorders or the condition that a human being is mainly suffering from that can't be treated or cannot be objected or subjected for the treatment, are the list of certain drugs coming under the Schedule J. Okay, so B is the correct option. According to the DPCO is mainly coming in the year of 2013. The scheduled formulation means any formulation include in schedule of the act. Any formulation is include in the schedule. In which schedule of the act? So Drug and Cosmetic Act is also having its two schedules, first and second schedule. The second schedule is related to the standard, related to the manufacturing, sale, distribution of the drug. The first schedule is mainly related to the books, related to ASU system. Yes, answer this question. I am just giving you time in order to solve these questions because I am giving you more information. Okay, because I want you to gather more information from a single question. You get around, I think, 5 to 10 concepts at a single time get cleared. So this is the way you can easily be able to learn all the things. So yes, a schedule does will mainly comes under the first schedule of the given DPCO Act 2013. Okay. So maximum retail price, that is also known to be as the MRP in our daily language or in our common language of the scheduled formulation is being fixed by on the basis of pharmacoeconomics. Okay. This is also responsible for defining the price of the essential drugs. So it is the ceiling price with the local taxes, the retail price with the local taxes, not the retail price, the average price, not the average price. The ceiling price is the highest price for the drug with the local taxes is mainly the maximum retail price. So A is the correct option. Okay. Coming towards our next question, any person aggregated by any notification issued regarding the DPCO 2013 may be applied to the government for a review within how much period of time if the government is already being published about him in the official gazette. So yes, so in most of the cases, this time period will mainly lies 30 days or we can say 1 month. Okay, in most of the cases, in case of licensing as well. Okay, so it is 30 days. Coming towards our next question, which chapter of medical device rules deal with labeling of medical device? That is related to the labeling of medical device. So there are multiple L's in the labeling. Okay, so L can be written in that way. Okay, L can be written in that way. So somewhere it is what? Somewhere it is what? It is 6. So yes, the chapter 6 of the medical device is mainly related to the labeling of medical devices. Coming towards our next question, the rule 36(1) of the medical device rule 2017, the Central Licensing Authority may grant an import license within dash from the date of application. Yes, within the within the 90 days of application, the Central Licensing Authority have to grant the import license for the drug for the medical devices. That is come under the rule 2017. Okay, that is mainly said under the rule 36(1), not the 19 days. Sorry. 90 months, not 9 months, 9 months. Sorry, 9 months. Okay, 9 months. Everyone starting answering. Should be somewhere in my mind. We are saying 9 month, but somewhere the 9. The medical device rule 2017. The retention fees shall be paid before the completion of 1 year from the date of issue, 3 years from the date of issue, 5 years from the date of issue, or 10 years from the date of issue. The retention fees that you have to pay before completion from the date of issue. Because the given license being assigned or being given for a period of 5 years, so before completion of that particular 5 years, you have to just pay the retention fees. Okay, so fees are answer. Coming towards our next question, according to the NDCT rule 2019, if the ethics committee concludes that a participant requires the investigational drug post-trial for the serious and life-threatening condition, who is responsible for providing it? So who is mainly going to provide it for the investigation of a new drug post-trials? So who is mainly responsible if anything will be happen? Either the central government? No, it is only giving them to approval. The principal investigator? No. The state health department, it is not related to it. Only the sponsor under which or who is mainly producing the given drug, the owner of a company, we can say, is mainly responsible for anything that will be happen in relation to that drug. Okay, so C is the answer. According to the New Drug and Clinical Trial Act 2019, which chapter deal with the import of new drug and investigational new drug for examination, test, and analysis? The NDCT Act 2019. Which chapters mainly deal with the import and investigation of a drug, of a new drug for examination by the examination, testing, or analysis? This is the last question of our discussion. The investigation and import of a new drug under the NDCT Act or the New Drug Clinical Trial Act 2019 comes under schedule, or comes under chapter 9. Okay, this comes under the chapter 9. So B is going to be the correct option. I hope you guys enjoyed that particular session. All the questions we have discussed today and all the things that are going to be really very helpful for your Tamil Nadu drug inspector exam point of view. Thank you so much for giving your precious time. We'll see you in the coming sessions. Thank you so much and all the very best for your exam. Thank you all.
Hello. Hello, everyone. I think now my voice is audible. Hello. So sorry, it was a connection issue. So now it worked finally. Chalo, let's discuss the questions. We will be going to discuss questions from your microbiology part. So first question. A medium which is composed of, which is composed a chemically defined compound is known as natural media, synthetic media, artificial media, and all of the above. Yes, answer this. A medium which is composed of chemically defined compound is known as natural media, synthetic media, artificial media, and none of the above. So these are basically called as synthetic media. Okay, what it is called as synthetic media. Option B is correct answer. Very good. Next question. Which of the following cultural media is used to provide a selective growth of gram-negative intestinal bacteria such as Salmonella and Shigella? Okay. Which of the following culture media is used to provide a selective growth of gram-negative intestinal bacteria such as Salmonella Shigella? Yes, your option is fluid thioglycolate medium, nutrient agar medium, 𝘂𝙀𝙎𝙎 agar medium, या soybean acid digest medium. Yes. So gram-negative intestinal bacteria are basically grow in your 𝘂𝙀𝙎𝙎 agar medium. Okay. So option C is correct answer. Okay. Chalo, next question. The principal structural component of the cell wall in bacteria is made up of. The principal component of the cell wall of the bacteria is made up of simple protein, glycoprotein, peptidoglycan layer, या complex polysaccharide. Yes. What bacterial cell consisting of? So when we talk about bacterial cell, it is basically consisting of peptidoglycan. Okay, peptidoglycan. And peptidoglycans are consisting of NAG and NAM, N-acetyl glucosamine and N-acetyl mumaric acid repeated unit. Okay, so option C is correct answer. Next question. The most active stage in the sequoidal curve of bacteria, which provides you maximum growth, which provides you maximum growth. Yes, answer this question. Maximum growth. Okay, so if we will see if you remember the curve, so it is starting first is your, so this curve is like this. So first is your lag, then log, then stationary, then decline. Okay, so here maximum growth is at this phase, that is your log phase. So option C is correct answer. Next question. Which of the following is not a component of peptidoglycan? Which is not a component of peptidoglycan? N-acetyl mumaric acid, N-acetyl glucosamine, teichoic acid, या tetrapeptide? Yes, answer it. Not a component of peptidoglycans. So teichoic acid, okay? So peptidoglycan contains N-acetyl mumaric acid, N-acetyl glucosamine, okay? And tetrapeptide also present. So, but teichoic acid is not present. Okay. Chalo. Next question. The absorption of poliovirus from GIT tracts takes place via pinocytosis, passive diffusion, phagocytosis, and none of the above. The absorption of poliovirus vaccine. So poliovirus vaccine from your GIT tract, how it will basically activated. Pinocytosis, passive diffusion, phagocytosis, and none of the above. Polio vaccines are absorbed through pinocytosis process. Okay, which process? Pinocytosis. Chalo, next question. Which of the following is not true for yeast? Okay, which of the following is not true for yeast? Yeast are eukaryotic, yeast are multicellular, yeast are reproduced by sexual and asexual process, yeah, none of the above. Yes, phagocytosis is cell eating, phagocytosis is cell drinking process. Very good. So, which is not true about yeast? So, first thing is yeast is a eukaryote. Yeast is a eukaryote. So, first point is correct. Yeast are multicellular. No, yeast are basically unicellular. Okay. So, option B is correct answer. Okay. Sure. Very good. Next question. Which of the following is an anaerobic media? Okay. Soybean casein digest media, fluid thioglycolate media, eosin methylene blue agar media, या mannitol salt agar media. Yes, answer this question. Which of the following is a anaerobic media? If you talk about anaerobic media, so fluid thioglycolate medium is a part of your anaerobic medium. Okay. Chalo, next question. Which of the following is a measure of total lethality of a heat sterilization process for a given microorganism, and it is used to compare the lethality of different heat sterilization process? Yes, F value, Z value, D value, या H value. So this is known as F value. Okay, what we call F value. F value. Okay, F value. Chalo, next question. Which of the following method would be appropriate for the sterilization of heat labile antibiotic solution? Heat labile. Okay. Dry heat using hot air oven, UV radiation, filtration through membrane filter, या gaseous sterilization using ethylene oxide. देखो heat labile, that means these are heat sensitive. Antibiotic solution, when it is come into the solution, we use filtration process. Okay, so option C is correct answer. Next question. As per the WHO, the percentage of ethanol in a alcohol based on hand rub disinfectant solution. What percent of ethanol should be there in a hand rub disinfectant solution? 60%, 90%, 70%, or 80%? When we talked about uh, hand rub disinfectant, 80% is basically used. Okay, 80% B by B. So option D is correct answer. So 70 to 80. Okay, actually the range is 70 to 80%. Okay, this range basically used. Okay, so this range. So next question. Which of the following statement regarding adjuvant is not correct? Okay, they increases the antigen specific immune response. They decreases the antigen specific immune response. They preserve the antigen integrity. They produce high affinity immune response, which is not correct. So what is adjuvant? So adjuvants are the chemical compounds which is given along with the vaccines to increase the response. Okay. They increases the antigen specific immune response. Yes, correct. They decreases the antigen specific. No, they do not decrease. They increase. That's why option B will be the correct answer. They preserve antigen integrity. Yes, they produce high affinity immune response. Chalo, next question. Which of the following T cells are capable of recognizing and killing the infected abnormal cell? Induced T cell, cytotoxic T cell, helper T cell, or suppressor T cell? Which of the following T cells are capable of recognizing and killing the infected or abnormal cell? So they are basically cytotoxic T cells. Okay, cytotoxic T cells. Next question. Which of the following is not true about double antibody sandwich ELISA procedure? Which is not true? Antigen absorbed to the wall surface. Enzyme labeled specific antibody is added which binds to the antigen. Antibody antigen antibody sandwich is formed. Degradation of enzyme substance produces a color image, which is not true about sandwich ELISA. Antigen absorb on the cell surface? No. Antigen antibody complex. Antigen antibody complex absorb on the cell surface. Okay. So option A will be the correct answer. Next question. Which of the following is not true for the immunoglobulin G? Okay, which is not true for immunoglobulin G. The most common and account for 80% of the immunoglobulin is serum. It is subdivided into IgG1, IgG2, IgG3, and G4, capable of passing from mother to fetus via placenta, exist in the pentameric form. Tell me which is not correct. Which of the following is not true for immunoglobulin G? So immunoglobulin G, it is exist in pentameric form? No. Immunoglobulin G does not exist in pentameric form. Who will answer? Immunoglobulin G exists in which form? Immunoglobulin G exists in which form? Comment down your answer in the comment section. Chalo, next question. Next question I am discussing. You comment down the answer in the comment section. Which of the following statement is correct about vaccines? Okay. Vaccine provides a immediate immunity. Okay. Vaccine contains a preformed antibody. Vaccine produced provides a long-term immunity by trigger the immune response. Vaccines are derived from the blood plasma. Tell me which statement is correct. Very good. IgG is existed in G existed in monomeric form. So vaccine provides a immediate immunity? No, vaccine does not provide you immediate. It takes times to produce it. Vaccine contains preformed antibody? No, vaccine contains either live or killed bacteria or virus. Vaccines are derived from the blood plasma? No. Vaccines are provides you a long-term immunity by triggering the immune response. So option C is correct answer. Next question. Which chemical is used to kill the Bordetella pertussis in the preparation of pertussis vaccines? Okay. Salicylic acid, acetic acid, formalin, या chloroxylenol. Which chemical is used to kill the Bordetella pertussis in the preparation of pertussis vaccine? Salicylic acid, acetic acid, formalin, या chloroxylenol. So which chemical is used? Formalin is used. Okay. Chalo, next question. Oral vaccines such as 𝘎𝙬𝙎 या 𝑠𝑎नcoral, they provides a protection against pneumonia, Ebola, cholera या polio. 𝘎𝙬𝙎 and 𝑠𝑎नcoral. Okay. So they will going to provide you protection against your cholera. Okay. So cholera, what is the causative organism of cholera? Answer in the comment section. Causative organism. Answer in the chat box. Which is the causative organism of cholera? In the chat box. Next question. Which pair consisting of WHO recommended malaria vaccine for the children in malaria endemic setting? RTS and R21, BCG and MMR, IPB and OPB, या HPV या hepatitis B? Yes. Which pair? That is your RTS and R21. So option A is correct answer. Next, which of the following test organisms are used in the microbial assay of erythromycin? Micrococcus luteus, Bacillus pumila, Saccharomyces cerevisiae, या Staphylococcus aureus? Tell me. Microbial assay of erythromycin. For microbial assay of erythromycin, we basically use Micrococcus luteus. Okay, Micrococcus luteus. Chalo, next. The creep in creep beer test, the inoculum is much heavier than the standardized inoculum. What is the most likely result? Large inhibition zone, smaller inhibition zone, no bacterial growth anywhere, yeah, complete sterilization of agar. In Cribe Bureau test, the inoculum is much heavier than the standardized inoculum. So what is the most likely result? The most likely result is it will produce small inhibition zone. It will going to produce small inhibition zone. Chalo, next question. A pathogen, a pathogen contains RNA as its genetic material. Which technique is required before conventional DNA PCR amplification for its genome? Okay, RNA contains as a genetic material. If the RNA is genetic material, which specific? Reverse transcriptase, gram staining, southern blotting, or preparation, protein precipitation. If the genetic material is RNA, we use RT-PCR. Okay, that is your reverse transcriptase PCR. So option A is correct answer. Chalo, next question. A plasmid carrying multiple antimicrobial resistance gene that is transferred through a direct bacterial contact. The mechanism is transformation, transduction, conjugation, or mutation. If it is carried out through a multiple antimicrobial resistance protein, so which technique is, so this is basically transferred through a virus. Okay, if it is transferred through virus, which technique we going to call it? We will going to call it as a transduction, because in case of transduction, the gene basically, in case of transduction, the gene basically transferred through a help of a cytoplasmic bridge. Okay, cyto bridge. Okay, cytoplasmic bridge. If it is transport, this method is called as conjugation. Okay, chalo, next question. Which antimicrobial susceptibility method directly provides a estimate for minimum inhibitory concentration from a concentration gradient? Gram staining, E-test, catalase test, or oxidase test? Yes, answer this question. Which antimicrobial susceptibility method directly provides an estimate of the MIC from a concentration gradient? Gram staining, E-test, catalase test, or oxidase test? It is basically transferred through your E-test. Okay. Chalo, next question. Which specimen generally considered more representative of a lower respiratory tract infection? Saliva, nasal swab, deep quality sputum, या oral rinse? More representative of a lower respiratory tract. When we talked about lower respiratory tract, your sputum is one of the more accurate things. So option C is correct answer. Next, which standard specifically address the quality and competence of a medical laboratory. ISO 9001, ISO 15189, ISO 14001, yeah, ISO 45001. If it is talk about quality and competence of the medical laboratory, then ISO 15189 is considered as the compatible with this. Next question. A patient develops a fever, abdominal discomfort, relative bradycardia after returning from an endemic area. Which organism is most likely to be caused? Salmonella typhi, Vibrio cholera, Shigella, या Campylobacter jejuni? Yes, answer this question. So first is fever, abdominal discomfort, and relative bradycardia. So this is a sign of typhoid. This is a sign of typhoid. So option A is correct answer. Next, malignant otitis externa may lead to which of the following complication? Cranial nerve palsy, retinal detachment, optic neuritis, या corneal ulceration. What it will going to cause? Cranial nerve palsy, retinal detachment, optic neuritis, या corneal ulceration. It will lead to cranial nerve palsy. Okay, it will lead to cause cranial nerve palsy. Okay, chalo, next question. A patient represent present with fever without an identifiable source despite of appropriate initial investigation. The clinical condition is best classified as non nosocomial infection, pyrexia of unknown origin, opportunistic infection, या latent infection. A patient present without an identifiable source despite of initial investigation. The clinical may be pyrexia of unknown origin. Okay, pyrexia of unknown origin. So 94B. 95. Conductive hearing loss is in acute otitis media is associated with accumulation of fluid in the middle ear, destruction of cochlear hair cells, cochlear nerve degeneration, या auditory cortex infraction. Tell me. Conductive hearing loss in acute otitis media. Conducting hear loss in acute otitis media is basically occurs due to fluid accumulation. Okay, fluid accumulation in the middle ear. So option A is correct answer. Next question. Which of the following terms specifically refers to a infection involving both renal pelvis and the kidney? Okay. Following terms specifically refer to an infection involving both renal pelvis as well as kidney. Cystitis, urethritis, pyelonephritis, या prostatitis. A condition where your renal pelvis and your kidney both are affected. Yes, answer the question number 96. It is due to pyelonephritis. Okay, pyelonephritis. So option C is correct answer. Next question. A major transmission route for pulmonary tuberculosis. This is very direct and most commonly asked question. Common route for the major transmission route for pulmonary tuberculosis. Airborne droplet, mosquito bite, contaminated urine, या animal bite. No animal bite involved, no contaminated urine, no mosquito bite. Air droplet. Air droplet from an infected individual, if they come in contact with a healthy individual, it will cause this condition. Next, a food poisoning episode with a very short incubation period and prominent vomiting It is a more suggestive of food intoxication, food infection, intestinal parasitic infection, या chronic food allergy. So short incubation period with prominent vomiting is a sign for food poisoning. That is your food intoxication. Okay. Next, which organism is classically associated with bacillar dysentery? Shigella, Vibrio, Giardia, या Candida? Which organism classically associated with bacillary dysentery? Shigella, Vibrio, Giardia, या Candida? Bacillary dysentery is basically associated with your Shigella. Okay, it is associated with Shigella. Next question. A woman present with thin grayish vaginal discharge with a fishy odor, vaginal pH is above 4.5, clue of cells are present. The diagnosis is vulvovaginal candidiasis, bacterial vaginosis, trichomoniasis, या gonorrhea. Yes, answer this question. Question number 100. It is a sign of bacterial vaginosis. Okay, it is a sign of bacterial vaginosis. Okay, chalo. Next unit. So now we will be going to talk about some part of your pharmacognosy. Okay, for the treatment, homeopathy tries to match drug picture to patient picture, drug picture to symptom picture, drug picture to chemical structure, drug picture to related drug picture. Yes. For homeopathy treatment, what we do? So we try to match the drug with symptoms. Okay. In homeopathy, we treat symptoms. So option B, drug picture to symptom picture. Next question. On the basis of raw drugs can be divided on the basis of taste. Raw drugs can be divided into how much category? 6, 5, 3, या 4. On the basis of taste, raw drugs can be divided into how much category? 6 category, 5 category, 3 category, or 4 category. On the basis of taste, they are divided into 6 category. Okay, they are divided into 6 category. Next, the pharmacopoeia in which the crude drugs are arranged according to their Latin title is British Pharmacopoeia, United States Pharmacopoeia, British Pharmaceutical Codex, या European Pharmacopoeia. Yes, the pharmacopoeia in which crude drugs are arranged according to the Latin titles. In which pharmacopoeia the crude drugs are arranged in a Latin title? These are basically your European pharmacopoeia. According to the European pharmacopoeia, they are arranged in a Latin title. Next question. Explain in plant tissue culture. Explain. Sorry, not it is explain, it is explant. Explant in plant tissue culture refers to tissue used to subculture from the previously cultured cell. Any part of the plant that is used to establish a new culture, whole seed used to initiate the culture, the entire plant from which culture material are sourced. So explant is nothing but these are the part of plants. These are part of plants from which a whole organism can be generated. A whole organism can generated. Okay, so option B is correct answer. Next question. Which of the following chemical test is used for the detection of tropane alkaloid? Van Wood test, Vital-Morin test, Lieberman-Burchard test, या nitrous oxide test? Which of the following chemical test is used for the detection of propane alkaloid? For propane alkaloid, what we use? We use 𝘂𝙀𝙎𝙀 test. Okay, 𝘂𝙀𝙀𝙀 test. So option B is correct answer. Next question. An alkaloid which is used as a local anesthetic is procaine, benzocaine, cocaine या lignocaine. An alkaloid that is used as a local anesthetic. An alkaloid that is used as a local anesthetic is your cocaine. Okay. Cocaine is used as a local anesthetics. Okay. Chalo, next question. Identify the herb that is used as the main source of atropine and scopolamine. Okay. It is used as the main source of atropine and scopolamine. Your option is Duboisia, colchicine, Nux vomica, या cinchona. The correct answer is Duboisia. Okay. Chalo, next question. Bromohexene hydrochloride is chemically synthetic derivative of. Bromohexene hydrochloride is chemically a synthetic derivative of vaccine, a synthetic derivative of bromine, a synthetic derivative of punarba, या natural वसा. Bromohexene is a synthetic derivative of vaccine. Okay. Chalo, next question. Modified anthraquinone of this test is specific for C-glycoside, O-glycoside, N-glycoside, या none of the above. Modified anthraquinone. Modified anthraquinone for this test is specific used for C-glycoside, O-glycoside, N-glycoside, या none of the above. Modified anthraquinone test is specifically used for C-glycosides. Okay. Chalo, next question. Killer Kiliani test. It is mainly performed for digitoxose, digitoxin या digitoxygenin. Yes, Killer Kiliani test is basically used for your digitoxose. Chalo, next question. Glyceragine, a sweet principle of liqueracy, each of potassium and magnesium salt of glyceric acid, sodium magnesium salt of glyceric acid, sodium calcium salt of glyceric acid, या potassium calcium salt of glyceric acid. What is glyceragine? Yes, answer this question. Glyceragine acid is a potassium and calcium salt of glyceric acid. Okay. Chalo, next. Tribulus tercetris is the botanical name of Gokru, Ganda Prasini, Tuvarka, या Pasanveda. Tribulus tercetris. Botanical name of Gokru. ठीक है, very good. Next question. Gingiberani is a chemically sesquiterpene, monoterpene, steroid या diterpene. Gingiberine is chemically sesquiterpene, monoterpene, steroids या diterpene. Gingiberine is a sesquiterpene. Why it will be a steroid? It will not be a steroid. Okay. Chalo, next question. Match the things. Okay. Clove is a clove active constituent is eugenol. Everyone knows. Next is phenyl. Phenyl active constituent is anethol. Eucalyptus oil. The active constituent is sinoil. Peppermint oil. The active constituent is methanol. So option D will be the correct answer. Chalo, next question. Eugenol is present in one of the following crude drug. Eugenol. Phenyl, tulsi, cardamom या coriander. Eugenol. So all you know कि clove contains eugenol. Other than the clove, your tulsi also contain eugenol. Okay, your tulsi also contain eugenol. So option B is correct answer. Next, gold bitter skin test is used to identify glycoside, tannins, alkaloids या flavonoids. Gold bitter skin test is used for the identification of glycoside, tannins, alkaloids या flavonoids. It is basically used for the identification of tannins. Okay, for the tannin identification. Next question. Which of the following drugs show hypoglycemic action? 𝘿𝙄𝙎𝙎𝙄, Asoka, Pterocarpus या black catechu? Hypoglycemic action or anti-diabetic activity. Sorry, diabetic anti-diabetic property. 𝘿𝙄𝙎𝙎, no. Asoka, no. Pterocarpus, so anti-diabetic activity. Specific. Chalo, next question. The pungency of ginger is due to the presence of the pungency of ginger, gingerol, gingiberine, sogal या curcumin. The pungency of ginger. The pungency of ginger is basically due to gingerol. Okay, it is due to gingerol. Okay, next question. All of the followings are used of Google except anti-rheumatic activity, hypolipidemic activity, anti-diabetic activity या anti-inflammatory activity. Use of Google except Google has anti-rheumatic activity, hypolipidemic activity, and anti-inflammatory, but it does not have anti-diabetic. So Pterocarpus have your anti-diabetic activity. Okay. Next, last question. If the resin contains benzoic acid or cinnamic acid, they are called as colophony, balsam, glycoresin or resin. If the resin contains acid or cinnamic acid, benzoic acid and cinnamic acid. So the correct answer is balsam. Okay, balsam. So that's all from my side. If you have any questions, so please ask. Otherwise, the session will be end here. So that's all for today. Thank you. Thank you so much.
Good evening everyone. So hope you have written your model paper test 2, right? So non-pharma already it is over. Now we are discussing the pharma core subject paper. Okay. So already you have completed some of the Sutix as well as the �々𝙎𝙎 sir class also. Okay. Now let us go to the analysis section. Okay. So in this analysis section you have observed the question related to the radioimmunoassays. So in this radioimmunoassays, let us see what are the questions you have faced. The decay constant, question number 121. The decay constant of a radioactive substance is related to its half-life by which formula? So this formula is not only the decay constant related to radioactive isotopes. In order to calculate the shelf life of the products also we are going to use this. So as you all know very well, that is 0.693 by T half. T half, it is very important, right? Whenever an isotope which is going to be injected into our body, so it should have a sufficient shelf life. So in order to calculate the decay constant for that particular radioisotope, whenever it is going to be injected in our body fluids, the formula we are going to use to calculate that. Okay, not only the decay constant, we can also call it as a shelf life of the product also. Okay, so nothing but what is the right option here? B equal B. That means lambda equal to 0.693 by T half. Okay. Okay, next question is also related to this radioisotope only. Which radioisotope is widely used in sterilization as well as radiotherapy applications? Mostly in the sterilization, whether it has done perfectly or not, and thereby radiotherapy applications also we are going to use it. So in these options, if you observe cobalt-60, iodine 125 and technetium 99, M phosphorus 32. So if you observe this phosphorus 32 means mostly in case of the DNA, RNA. So labeling of these DNA and RNA, we are going to use this phosphorus 32, and it has a T half of I think 14 days, right? So more than 14 days it is going to be there, 14 to 15 days in between that. So technetium 99M, actually this radioisotope, it is going to be used in order to see the after the diagnostic therapy. Okay, in order to see some of the internal organs like heart, bones and all these things. In order to observe those internal organs, we are going to give this radioisotope. Okay, and as you all know, iodine 125, iodine 131, all these are going to be used in case of the thyroid hormones. And in case of the sterilization, okay, we are going to use this cobalt-60. Okay, so it will give us the information related whether the sterilization has completely done or not. Okay, next. Which one of the following provides the best protection against the external gamma radiations? Electromagnetic radiations, very fast if you see the cosmic rays, then gamma rays. So in that gamma rays also, how we are going to protect that? Okay, so which one of the following provides the best protection? They are asking best protection. Paper sheet, aluminum foil, lead shielding as well as cotton gloves they have given. As you all know, these three, A, B and D, are not at all going to stop the gamma rays, because they are going to have very high energy. Gamma rays are there. Okay, but only the any type of the compound which is covered with the lead coating. Okay, so lead coating can stop the penetration of not only gamma rays, X-rays also. X-rays also can be stopped by taking the help of this lead coating. Okay, so that's why option C it is the right answer. Next question, which of the following is not a recommended principle of radiation? Okay, so sequentially they have given the questions related to radioisotopes only. Very good. Which of the following is not a recommended? Not is very important here. Recommended principle of radiation protection. So whenever we are going to see some of the CT scans, XRDs, X-rays, scannings are going to be done, they don't allow us inside except the patient. Okay, because the radiation energy it is going to be more there. So here what they are asking, which is not a recommended principle. That means which is not acceptable one, we need to find out. Minimize the exposure time. Actually this is recommended. This is not recommended means it is not the suitable answer. Always try to minimize the exposure time. That's what we need. Next, maximize the distance from the source. Yes, that is also the right option, the right way in order to protect from the radiation. Okay, so that's why in that case this is also not suitable. Use appropriate shielding. Just now we have seen the lead shielding, how we are going to, so this is also right. That means this is also a recommended procedure. Next, increase the exposure time to obtain the better results. No, nowhere it is going to be recommended. So this is not the recommended procedure or principle in order to protect it from the radiations, any type of the radiations. Okay, so what is the right answer here? Option D. Yes. So with your minimum common sense you can guess that answer. Next, which impurity may arise due to interaction between the drug substance and the container? That means sometimes the drug, it is going to be getting absorbed on the container walls also, right? So here the impurity may arise due to the interaction between the drug substance and the container. Because of the container, whatever the vessel we are going to store, or whatever the vessel we are going to transfer some of the drugs, so all those are also should be sufficiently inert substances. They should not have any reaction. Okay, so here if you observe, manufacturing impurity, container related impurity, deliberate adulterant, atmospheric impurity. So container here it is there. So definitely you are going to select the option B only. So whenever they are talking about the container means, obviously we are going to choose the option B only. No one can select the other answer, right? Okay, next. In the limit as for the chlorides, okay, limit as for the chlorides, turbidity is produced due to the formation of, we know that chlorides are going to be estimated by using the reagent silver nitrate, right? So whenever chlorides are present, you are going to see the formation of silver chloride, which is a precipitate. That means it will give, if ppm levels it is going to be there, means we are going to observe the turbidity of the solution. Not only chlorides are present, even if you take the tap water also, if you just add the silver nitrate solution to take the silver nitrate solution, add 1 drop of tap water, you will see the turbidity, because chloride ions are going to be present in that. So here, yes, silver chloride, due to the formation of this silver chloride. Okay, so this is a right option. Next, which factor is most likely to cause a gross error in the volumetric analysis? In the errors we have so many types of the errors, random errors, okay, as well as personal errors are there, systematic errors, methodological errors, all these are. Gross error means completely total error. How we are going to see that? Let us see. It is going to be in the volumetric analysis, which is going to cause the gross error. Slight temperature fluctuations, misreading the burette. That means burette, if you don't see without parallax error, you have to take the burette reading. So misreading the burette due to the incorrect observation. Yes, this is going to be the one of the option. Random variation in the endpoint means you are going to observe the random error. Small instrumental uncertainty means systematic errors you are going to observe. Okay, so temperature fluctuations means environmental factors are responsible for the errors. So gross error means we are going to see because of this entire, the person who is taking the wrong reading, okay, from the burette. So if you take the wrong observations and all these things, then you are going to observe this gross error. Okay, next, which statement regarding accuracy and precision is correct? Mostly till now whatever the questions we have seen, all those seems to be very easy. I think everyone has given the right answer, right? It looks somehow easy today in this model question paper 2. Analysis subject is seem to be somewhat easy questions you got. Okay, 128 question, let us see. Which statement regarding the accuracy and precision is correct? Which one is the correct option? Let us see the options first. Accuracy refers to the closeness among the repeated results. Whenever repeated it is going to come means we should remember the word precision. Repeatability, that is related to the word precision. So this is not related to accuracy. Precision refers to the closeness of the true value. Actually this is not precision, this is called accuracy. Accuracy means which is closeness, nearness to the true value. Okay, so these two are the Wrong answers. Accuracy refers to the closeness to the true value. Yes, this is the right option. Accuracy and precision have identical meanings. No. These two are independent each other. Okay. Accuracy means which is nearest to the true value. If your acceptance criteria is 100. So if you are getting 99, 98, 97 like this, which are about nearer to the true value, means those are called accurate results. So if you get consistently the same results, 97, 6 times you got the 97 or 6 times you got 87. Like that, if you get the repeatable results, that is called as precision, right? So in this case, what is the right answer? C. Accuracy refers to the closeness to the true value, right? Okay. Which of the following is a secondary standard? Secondary standard means what actually? It is not as pure as that of the primary standard, and it is having some environmental factors like hygroscopic, deliquescent, okay, like that some of the properties are going to change with the atmospheric conditions. Such type of the compounds we can use as primary standards, secondary standards in the titrometric analysis, but they need to be standardized against the primary standards. Okay, so if you see the options they have given, they are asking secondary standards. Potassium hydrogen phthalate, PHP, it is a good primary standard. Sodium carbonate, this is also a good primary standard. Sodium hydroxide, this is actually hygroscopic in nature, so the strength, it is not going to be stable at the room temperature, so that's why it is going to be one of the secondary standard. Oxalic acid dihydrate, this is also primary standard. It is also very highly stable compound. So what is the right answer here? Sodium hydroxide. Okay, next. Match the following. Yeah. Aluminum hydroxide, zinc oxide, sodium fluoride. Yesterday in model paper 1 also we have seen the questions related to this. Okay, as we know that zinc oxide, it is going to be used as a astringent as well as protection agent, which is going to be used in most of the powders are going to be, skin powders we are going to use it. So 2B, it is the right answer. So if you see the options, in the first option A itself, 2B it is there. So 1A, aluminum hydroxide, it is going to be used as antacid. Sodium fluoride, it is going to be used in the tooth caries. That means as an anti-tooth carie agent, it is going to be used. Okay, so in the dental problems. So 1A, 2B, 3C, direct straight matching, no jumbling at all. Okay. Which inorganic compound acts as an antacid and adsorbent also? Both properties it is going to possess. Which one it is going to possess that? Magnesium trisilicate, potassium chloride, sodium fluoride and boric acid. Actually, if you these three are there, these are not going to be used as adsorbents, right? So not even antacids also. So if you observe this magnesium trisilicate, which is going to be used as an adsorbent in the column chromatography also, as well as in some of the normal therapeutic applications also, we are going to use this, as well as an antacid also. Okay, so option A, it is the right answer. So how many have given the right option? Okay, Aspirant Pharma. Okay, very good. Definitely, I think everyone has given the good answers for the analysis. Very good. Which of the following is primarily used as a topical antiseptic? Topical antiseptic, as you all know, that is regular usage also. We are going to use the Dettol. All these some of the antiseptic soaps are also going to be Savlon. All these liquids we are going to use it. So in that, what is the content we should know, right? So as a pharmacist, we should understand what is the ingredient in that. So as an antiseptic topical agent, boric acid, magnesium hydroxide, sodium carbonate, bicarbonate, calcium carbonate. So if you see this, topical antiseptic, generally we are going to see that one as a boric acid powder. It is available in the form of powder, right? So whenever some cuts are there on the skin, we are directly going to add this boric acid powder, right? And generally it is going to be used in some of the other household purposes also. In order to store the food grains also, they are going to mix this boric acid in order to prevent from the flies and all these things. Okay, next. Statement related question. Primary standard should possess high purity. Definitely. Definitely primary standard first character is it should have the high purity, high molecular weight and all these things. Okay. The concentration of the primary standard solution can be accurately calculated from accurately weighed amount. Yes. Concentration can be calculated from the weight only. If you weigh the exact amount based on the anhydrous form or monohydrate form or dihydrate, whatever the form you are going to take it, based on the accurately weighed amount, according to that you can calculate the concentration of the primary standard. So this is the right answer. So both A and R are true and R correctly explains A. Okay. So everyone has put the right answer, right? Okay. Which of the following pairs is incorrectly matched? Pairs are incorrectly matched. Sodium bicarbonate, systemic antacid, magnesium hydroxide, antacid. These two are going to be antacids. We know that very well. Zinc oxide, skin protectant. Just now we have discussed. Yes, that is also right answer. Sodium fluoride, expectorant. Actually, sodium fluoride, which is going to be used in the tooth caries. Okay. So in that, as an anti-carrying tooth carrying agent, we are going to use this in the dentistry. Next, if a radioactive substance undergoes two half-lives are present, two T-half, whatever it is there, it is going to possess the two half-lives. Then what fraction of the original activity remains? Whenever any compound is having the two half-lives, then we can take the multiplication of these two half-lives. Okay. So automatically if you see the fraction of the remaining original means you can take only 1/4 only will remain. Two half-lives it is going to, first part it will 50% will be over, second part another 50% will be over. So in 50 again 50 means only 25% only remains in the whatever in the biological fluid. Okay. So that's why the option here it is the 1/4. Next. Question number 136. Yeah. Okay. In gel electrophoresis, molecules migrate under the influence of. In gel electrophoresis, actually we are going to use the different types of the gels. So in this case, they are going to have some electrostatic forces in order to bind the molecules. Which type of the molecules actually we are going to use that? Like proteins as well as amino acids, right? So here if you observe, the molecules migrate under the influence of which force? Magnetic field, electric field, gravitational field, thermal gradient. Actually if you see the gel electrophoresis, we are going to use the electrostatic force. No magnetic field, gravitation, all these are not going to be observed. Okay. So different types of the gels, either horizontally or vertically we are going to use it. Okay. So obviously option B it is the right answer. Electrostatic forces are going to be there. So thereby the separation it is going to take place. Okay. Calibration of an analytical instrument should be performed using appropriate reference standards. Okay. Calibration anyhow you are going to use like dissolution apparatus it is there. We are going to use the caffeine solution, right? So similarly, HPLC it is there. The calibration you are going to use the same caffeine solution, as well as UV spectrophotometer it is there. For the calibration, potassium dichromate it is going to be used, right? So different instruments are going to be used for the different reference substances. Okay, this is the right statement. Calibration establishes the relationship between the instrument response and known reference values. Yes, that's why we are going to use the particular substance as a reference standard if we know the values of that compound. So both A and R are true and R is the correct explanation. So here option C it is the right answer, right? Okay, everyone is giving the right answer. Very good. Okay, let us see the next question. 138. Gas chromatography is most suitable for the compounds. GC, when we are going to use this? Actually, the substances are volatile and they are different in the boiling points, right? And they are thermolabile also. In these three characters, if they fulfill, then we can estimate them by gas chromatography. Here, what are the options they have given? Highly ionic and non-volatile. Non-volatile means we cannot estimate by the GC. Volatile and thermally stable, yes. This is going to be the Right answer. Insoluble in all the solvents, no way. Always high molecular weight, no. High molecular weight polymers are not at all going to be used in GC. For that actually we will go with the molecular exclusion chromatography, size exclusion chromatography, right, where we are going to use a high molecular weight polymers. So in this case option B, it is the right answer. Next, the carrier gas in GC, because GC means gas chromatography, where the inert gases are going to be used, mostly helium gas is used as a mobile phase. So that's why here the gas it is going to be used and it is taking the analyte along with it. So we are going to call that one as the carrier gas. So here carrier gas in GC acts as mobile phase. Okay. Okay. Option B, it is the right answer. Okay, 140 again statement question. Temperature program is useful in the gas chromatography for the analysis of mixtures containing the compounds with wide range of boiling points. Yes, temperature program it is required, because when we observe the boiling points differentiation, then by doing the temperature programming, we can separate those two compounds or those whatever the mixture of compounds are there. So temperature programming can be done in the GC based on the boiling points of the compounds. Temperature programming gradually changes the column temperature also during the chromatographic run, right? Is it going to change the temperature of the column also? Yes, definitely. In the GC, we are going to see the everything, it is going to be under the controlled oven, the column as well as all these things. Okay. So that's why A and R both are true and R is the correct explanation. Next, specific rotation. Okay, regarding the polarimetry, the question is a characteristic property used for comparison and identification of optically active substance. Yes, yesterday also we have seen one question same as that of this. Yes, this is the right answer, right statement. Observed optical rotation depends on factors such as concentration and path length. Yes, this is also right answer. According to B.S. Lambert's law, how we are going to see the same here also. The optical rotation, we can determine the concentration by factors affecting the particular optical rotation or concentration as well as path length. So this is also right answer. P is right, Q is right. Next, temperature can influence the optical rotation of a substance. Yes, definitely, because if we are going to see the temperature, whatever we are going to see 20 or 18, you need to mention 20 or 25 degrees centigrade, right? Definitely the temperature, it is going to change the value of optical rotation. So this is also right answer. Polarimetry can be applied to determine the concentration of an optically active substance when appropriate conditions are established. Definitely, because optical rotation, this optical ORD curves are going to be there, right? So by determining the optical rotation, we can plot a calibration curve, and from that the unknown sample concentration can be determined. So this is going to be one of the useful technique if you just know that it is optically active. If the analyte it is optically active, then you can determine the concentration of unknown substance also. So all the statements are right. Okay. So P, Q, R and S. Yes. Option C, it is the right answer. Next, the endpoint in the electrometric titration, that means electroanalytical methods, right, can be detected by measuring changes in. So if the electrolyte is having the changes in any type of the electrochemical properties like potential, that means electromotive force, conductivity, like this, if you are going to observe, then definitely we can find them by electrometric titration. Here in the options they have given density, electric potential, refractive index and viscosity. So if you see this electric potential only, it is a property of electrochemical property. Okay, potential. So this is going to be estimated in terms of the EMF, electromotive force. Whereas if you see the density, refractive index and viscosity, all these are the physical properties, not the chemical properties. Okay. Okay. Next, fluorescence intensity may be affected by concentration. Okay. particularly when the solution is not sufficiently dilute. Yes, if concentration it is more, then fluorescence it is going to be getting decreased. That is called as self-quenching, right? So that's why appropriate concentrations are also responsible for giving the correct fluorescence. Yes, this is the right statement. Quenching can decrease the observed fluorescence intensity. Yes, this is also right answer. In quenching, we will observe so many types. Chemical quenching, kinetic quenching, like that. Self-quenching. Next, fluorescent derivatives may be prepared when the original analyte has weak fluorescence. Okay, derivatization. So, derivatization can be done so that the fluorescence can be increased. Okay, that is also possible. So, P is right, Q is right, R is also right. Next is, every compound that strongly absorbs UV radiation necessarily exhibits strong fluorescence. No, this is wrong answer. If it exhibits the UV radiation, that means the electrons are getting energy and excited to the excited state. N electrons, pi electrons, sigma electrons are going to be involved. But it is not needed that they will exhibit the fluorescence also while coming back to the normal state. That is not required. Okay. So, S is wrong. Remaining 3 are right answers. So, if you see the options, C, P, Q and R only the right statements. S is wrong. Diazotization titration is primarily used for the compounds. Diazotization whenever we are going to use in case of the compounds which contain the primary amino group, which are going to be attached to the most of the aromatic compounds. Okay. So, in the options if you see, primary aromatic amines, carboxylic acid, hydroxyl group, ketone group. So, obviously option A is the right answer. So, in the diazotization actually what is going to happen? That is N double bond N, diazo compound. So, it is going to be salt, means diazonium salt it is going to be formed, right? So, that is why we are going to use a amino group. Whenever it is going to be present, those compounds can be determined quantitatively. Next, in gel electrophorese, molecules migrate under the influence. Already we have seen this question, right? Oh, so you are lucky, 2 times you got the same question. Electric field, electrostatic forces are there. So, those two are the electrostatic forces are responsible for moving this, right? So, option B it is the right answer. Okay. The sample generally needs to be converted into free atoms before atomic absorption measure. Okay, atomic absorption spectroscopy we are talking about this. So, whenever we will give the energy, so those sample whatever it is there, it will be converted to the free atoms, right? So, that is also going to be the right option. Hollow cathode lamp can serve as a source of element specific. Yes, whenever you want to determine specific element, cobalt means you have to use a cobalt hollow cathode lamp. Copper means copper hollow cathode lamp, you have to use it. Specific elemental lamp source you have to use it. Yes, this is also right answer. Atomic absorption spectroscopy is particularly useful for elemental analysis. Yes, elemental analysis only, that too mostly the metals, okay, as well as nickel, aluminum, wherever you are going to see those such type of the metals, definitely we are going to see by them in the atomic absorption spectroscopy. That is also right answer. Next, the analytical wavelength is selected without regard to the element being determined. Without it is not the right answer. This is actually based on the element which we are going to determine. On that basis only we are going to select the lamp. From that only we are going to select the wavelength and all these things. So, S is wrong. So, only these 3 are right. So, P, Q and R. So, what is the option here? B, right? So, B option it is the right answer. Right? So, everyone, someone has put the wrong answer. Okay. Don't worry. Next, infrared spectroscopy is particularly useful. Yesterday also the same question we have seen, to determine the functional groups, right? By stretching and the bending vibrations, we are going to observe what are the functional groups present in the particular molecule. But here you need to know that in the IR spectroscopy, what is the source of radiation and what is the calibration standard. What is the calibration standard? Polystyrene, right? Polystyrene film, directly it is going to be used. So such type of the important points, and whenever IR it is going to be taken up by any molecule, it should cause the change in the dipole moment. Then only you are going to observe the IR spectra. Otherwise, no signal it is going to be observed. Okay. Next, the stationary phase in the TLC, thin layer chromatography, the stationary phases, so many are there: strong, medium, weak stationary phases like adsorbing materials, because in thin layer chromatography we are going to use the solid as the stationary phase material, right? And anyhow liquid is the mobile phase, we are going to use it. So in that, if you observe silica gel, all these three are the liquids, we don't use as the stationary phases. Only the silica gel, it is a widely used solid material which is used as adsorbent, right, in TLC. Next, perchloric acid is commonly used as, okay, HClO4 as a titrant for the assay of weakly basic pharmaceutical substances in the non-aqueous titration. Yes, perchloric acid, it is going to be strong acid. So, weakly basic compounds can be determined. This is the right statement. Perchloric acid is a strong acid, okay, in suitable non-aqueous solvents and reacts quantitatively with weak bases. Yes, this is also correct, somewhat not perfectly explaining, but 70% it is the right answer. Okay. So, both A and R are true and R is the correct explanation. Yes, option A, it is the right answer. So, no need to bother about the other options. Okay. Next, a blank solution is used in spectrophotometric analysis before measuring the sample. Okay, for the correction process, because sometimes we are going to use different types of the solvent mixtures are going to be used as diluents, right, in spectroscopy. For that purpose, a blank correction it is required. And the blank compensates for the absorbance arising from the solvent, yes, and other components of the analytical system. Yes, definitely. So, this is the reason, correct reason they have given. So, both A and R are true and R is the correct explanation. First option itself it is right. So, you can save more time, okay, in the examination. Next, a pharmaceutical laboratory is comparing TLC with HPTLC. Okay, TLC with HPTLC. Consider the following statement. Okay, let us see what are the statements. HPTLC generally employs the plates. Yes, TLC plates, they are going to use it with a finer and more uniform stationary phase layer. Yes, particle size. In case of the normal TLC, if you see, the particle size it is going to be 60 to 200 microns. But wherever in HPTLC, if you see, around 50 microns, less than 10 to, that means 10 to 50 microns or 15 microns are also going to be used. Particle size is going to be very important. Yes, this is the right statement. Automated sample application can improve the reproducibility of the HPTLC. So, that's what we are going to take the advantage of this HPTLC. Yes, this is also right answer. Densitometric scanning can permit quantitative evaluation of the separated zones. Densitometry, that means it is nothing but the HPTLC only. It has the another name of densitometric technique because of the fractions, the colored bands are there. Based on that, we are going to calculate the quantitative amounts. Yes, this is also the right answer. HPTLC eliminates the need for chromatographic development. Definitely it is required. Chromatography has to be developed. Chromatographic conditions are required. Then only you can use it in the HPTLC, right? So, option S is wrong. So, remaining 3 are right. So, here if you observe the option A, P, Q and R, first 3 are right, whereas fourth one it is wrong. Okay. Non-aqueous titration is particularly suitable for the assay of. When we opt for the non-aqueous titration, whenever the compounds are not giving the sharp endpoint, or sometimes if indicator it is not available. So, in which cases we are going to get all these problems? When our compounds are either weak acids and weak bases. Yes, In these two cases, we are going to go for the, we are going to select the non-aqueous titrations. Okay, strong, whenever even one of them it is strong, we will go with the aqueous titrations only. Okay, okay. Next, option B, here it is the suitable one. Refractive index, which is influenced by the wavelength of light used for the measurement. What is the light here we are going to use? That is sodium lamp. Okay, sodium lamp at 589 nanometer, yellow color light it is going to be used. That is the source of radiation. Temperature control is important in the refractometric measurements. Yes, definitely. Temperature is also very important. If temperature changes, the refractive index also angle will change. Refractometry can be used for assessing the identity or purity of the certain pharmaceutical substances and preparations. Definitely. Refractive index, it will give an identity, and if it is not pure, then refractive index value also changes. So this is also the right statement. Refractive index is completely independent. Here only Q statement it is depending on the temperature, and S is telling that independent. No, S is wrong actually, right? So first three are right. P, Q, R are the right statements. Got it? Yes. A, option A, it is the right answer. Okay. Next, in paper chromatography, the stationary phase is primarily associated with. In paper chromatography, we are going to use different types of the papers like Whatman paper also it is going to be used. Grades are available, right? So, in which we can see the principle of both partition as well as adsorption also, right? So, how, when we are going to see the partition as the principle, the pores are present in the papers. So, the water present in those fibers, it is going to be acting as a stationary phase. Then, the principle of separation is the partition. Whenever the papers are directly used, that means impregnated papers are there, like acid washed, base washed, silica impregnated papers, like that they are going to use it. So, in which case we will see the solid as the stationary phase, then adsorption it is the principle of separation. Okay, here what they are telling, stationary phase is primarily associated with cellulose and its retained water. Yes, these papers are going to be used. This is the right option. Nitrogen it is not used, vapor, mercury, no, only cellulose. Okay. So, if you hear the word cellulose itself, we can understand that is the right answer. Next, ferric ammonium sulfate is a stronger oxidizing agent in acidic medium than potassium dichromate. Yes, actually ferric ammonium sulfate, it is going to be somewhat more stronger oxidizing agent. That is true. Okay, statement 1 is true. Ferroin indicator, it is commonly employed in the serimetry. Yes, it is going to give the blue to red color. Okay, so the endpoint it is going to be observed. Whenever serimetry it is used, it is a suitable indicator. So both are true. Okay, so this is going to be the right answer, B. Next, erychrome black tea, it is commonly used for the estimation of erychrome black tea. It is a PM indicator. In complexometry we are going to use. So definitely in complexometry means metals are going to be estimated. Sodium, it is calcium, magnesium, chloride, sulfate. So in this if you see calcium and magnesium, whenever these are present, we will go with the complexometric titration. So option B it is the right answer. Next, which ICH guideline primarily deals with validation of analytical procedures? You know that ICH guidelines are available, Q guidelines, right? So from Q1 to Q10 these are all available. Okay. So in this if you see the guidelines related to analytical procedures, validation, not procedures, validation of the analytical procedures means we are going to see the guideline related to Q2. Okay. So whereas if you see the Q1 means it is related to stability guidelines, Q1 to Q1A to Q1F you are going to observe related to stability guidelines, and so this Q7 guidelines are also there, where we are going to see the like I can tell you that Q7 guidelines which are going to be used for the GMP, good manufacturing practice, okay, and Q9 it is going to be used for the quality risk management, okay. Not only up to 10, 14 is also going to be, Q1 to Q14 are also available, but Q7 especially for the GMP, okay, whereas 9 is for the quality risk management, we are going to use it. So validation purpose, Q2 it is a suitable answer. Next, the molecular ion peak in mass spectrometry can provide the information. Molecular ion peak, mass spectroscopy, what we are going to see, the graph it is going to see the M/Z ratio, mass to charge ratio. It is going to be taken on the x-axis and relative intensity it is going to be taken on the y-axis. So we will see the peaks related to that. So highest intensity peak it is going to be called as base peak, right? So whenever 100% peak, it is the tallest peak, whatever it is there. So here what they are asking, molecular ion peak, okay, molecular ion peak means M plus, whatever it is there, highest mass by Z ratio, heavy ion, whatever it is there, highest molecular weight, can provide the information about the molecular mass. Yes, this is going to give you the information related to the molecular mass, okay. Melting point, refractive index, optical rotation, no. These are not going to be obtained from the mass spectroscopy. Next, in differential thermal analysis, okay, thermal methods of the analysis we have studied in DTA. The analytical signal represents the difference in differential thermal analysis, the name itself you can come to know that there are temperature differences are going to be observed. The difference between the temperatures are recorded between the sample and the reference, okay. So here if you see the options, temperature between the sample and the reference, yes, exact words we have seen, okay. So this is the right option. Heat absorbed directly, heat absorbed or heat emitted, what is the heat content it is there, that is going to be observed as enthalpy, that is we are going to see in case of the DSC, differential scanning calorimetry, okay. And mass of the sample during the heating, that is going to be observed in the TGA, thermogravimetric analysis, we are going to concern about the mass of the sample, and absorbance of the sample at different wavelengths, this is related to spectroscopy, okay. So now what is the right answer? Option A, okay. Next, in gravimetric analysis, the precipitate should ideally have a low solubility. Yes, in gravimetry what is our aim? In order to get the mass of the compound, highly purified compound, by the gravimetric titration finally you should get the particular substance in the solid form. So definitely the solid, whatever it is going to be prepared by precipitation process, it should have a low solubility. Definitely it is the right answer, right statement. Low solubility minimizes loss of analyte during precipitation and filtration. Yes, while gravitization, that means gravimetric analysis, whenever we are going to do, we are going to follow some series of the steps, right. So sample preparation, addition of the reagent, then precipitation, then Ostwald ripening it is going to take place, and digestion is going to take place. Afterwards filtration step will be there, then drying, okay, weighing, then calculation. All these steps are going to be involved, but here you need to observe that low solubility of the precipitate always increase the yield of the sample, okay. So in this case both are the right options. So both A and R are true, and R is the correct explanation of A. Yes, so this is going to be the right answer, okay, fine. So these are the questions related to the analysis, okay. Now you can have any doubts, you can ask it, otherwise Devanshu sir will take the next session, okay. So from now onwards, the second part, the next part. Any doubts are there, you can ask. So thank you, students.
Okay, now am I audible, guys? I think now I'm audible. Please confirm me first. Am I audible enough? Yes, come on. Okay, now am I audible, guys? I think I'm audible. Okay. Okay, fine. Yes, just confirm me first. I think I'm audible. Yeah? All right, fine, 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 fine. Okay, so okay, guys, so I think this is going to be the last pharma section, Hana, in this video session for you guys. Hana, I know your session is too long, got too long. Okay, but now I'll be doing it fast, fast, fast. Hana, because you know the pharmacology questions will be definitely going to be easy for you, right? Hana, so just give me a thumbs up and then we can start the session. Just give me a thumbs up, guys. Yes, so that we can start the session now. Yeah, come on, guys. Just give me a thumbs up and then we can start the session. Yep. Okay, fine. Chalo, let's go ahead now. So here's the first question for you guys. That's from pharmacology. Which of the following drug is not come into the category of prodrug? Okay. Which of the following drug is not come into the category of prodrug? Levodopa, proguanil, becampenicillin, ampicillin. Levodopa, dopamine. Proguanil, cycloguanil. Becampenicillin will give you ampicillin, but ampicillin is not a prodrug, right? So ampicillin D is going to be the correct answer. Ampicillin ka prodrug would be your becampenicillin, simple. Now, which of the following teratogenic drug? Which of the following teratogenic drug may cause neural tube defects, right? From the part of your general pharmacology only, in which we have talked about that adverse effect, or you can say a teratogenic effect table, right? Teratogenic effect table. And over there, we do have neural tube defects. Okay, so isotretinoin, aspirin, carbamazepine, or progestins. What do we have? Isotretinoin, aspirin, carbamazepine, as well as progestins. What do we the correct answer, guys? Can you tell me? What should be the correct answer over here? Yes, come on. Yeah. What's going to be the correct one? Over here, that would be your carbamazepine. Okay. Along with carbamazepine, this neural tube defect is also associated with your sodium valproate. Okay, or you can say valproic acid, or you can say sodium valproate. Okay, isotretinoin, facial changes, right? Aspirin, pre-closure of ductus arteriosus, carbamazepine, progestins, virilization, right? All these things going to be takes place over here, right? Now let's go on to the next question. Carbamazepine is going to be the correct one. Now, match the following receptor with their example. Okay, inotropic receptor, GPCR, enzymatic as well as nuclear receptors. What's that? Inotropic receptors, GPCR, enzymatic or nuclear receptor. Simple. So, inotropic, transforming growth factor, T4, T3, 5-HT3, alpha 2 mu adrenergic receptor. Okay, both the receptors. Okay, fine. So GPCR, obviously it's going to be your alpha 2, because these are what? GI mediation, GDI mediation over here, right? Inotropic, it would be your 5-HT3, so 1 ka 3 and 2 ka 4, right? Enzymatic, this one, right? Growth factor, as I told you, pig cytokinin, if you remember, right? So 1, and this nuclear receptor would be your T4 to T3, that's your 2. So 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2. D is going to be the correct one. Yes, easy enough, right? Very good, Samyukta, Nurkatnam, and Majida. Yes, exactly. Aspirant, very good. D is going to be the correct answer. Very nice. Let's go ahead now. Okay, let's go. Yes. Which of the following is true for receptor action of a drug? Receptor action of a drug. An antagonist has both intrinsic activity and affinity for receptor. Antagonist, they are asking. Okay, antagonist. An antagonist has affinity but no intrinsic activity. A partial antagonist has no intrinsic activity. No, it has submaximal activity, right? It has what? Submaximal activity, right? Submaximal activity, right? Now, intrinsic activity and affinity are not important. Obviously, no. So B is going to be the correct one. Simple enough. B is going to be the correct one. Yes, easy, everyone. Easy enough. Yeah. Exactly. Very good. Majida and Sneha, Rupa, Amlan, very good. And Nurgatnam. Fine. So B is going to be the correct one. Now, uveoscleral outflow of aqueous humor is increased by pilocarpine, beta blockers, alpha agonist, or carbonic anhydrase inhibition. Yeah. Uveoscleral outflow is going to be increased by pilocarpine, beta blockers, alpha agonist, and carbonic anhydrase inhibitor. Yes, easy enough. Yeah, that also you need. Pilocarpine, beta blocker, alpha agonist, and carbonic and where is PGF2 alpha agonist, or you can say analog, bimatoprost, latanoprost. Where is that in option? This would be the correct answer, right? Over here, pilocarpine going to increase the aqueous humor. Uveoscleral outflow के through तो नहीं करेगा भाई. This is not going to act through uveoscleral outflow. Pilocarpine, however, it is going to, you can say, increase the trabecular outflow, I would say, right? It's going to increase the trabecular outflow? No, not trabecular outflow, but only outflow. Only outflow. It is a meiotic drug, right? It is a meiotic drug. Meiotics drug. This is not going to be the correct one. Latanoprost, PGF2 alpha, right? If you remember, PGF2 alpha analog, that is a latanoprost, bimatoprost, right? These are all the ones which are going to increase the one. Pilocarpine is not going to be the correct answer. No. Now this one, guys. Mechanism of action of carvedilol. So beta 1, beta 2, and alpha 1 blockade. Beta 1, beta 2, and alpha 1 stimulation produce vasodilation. Beta 1, alpha 2 block produce vasodilation, or none of these. Chalo, easy enough. Easy question. Yeah. Yeah. Easy enough, guys. Which is going to be the correct one? Tell me. Yes. Which is going to be the correct one? Over here. That's your A only. How? As I said, alpha 1. Alpha 1 is going to do what? Alpha 1 blockade produces vasodilation. Alpha 1 is going to do what? Alpha 1 have GQ protein. So if you're going to inhibit this one, vasoconstriction was happening at that time, but it will be doing vasodilation. Obviously, action of carvedilol will be acting onto the beta 1 and beta 2. You know that that's a mixed acting drug, alpha plus beta blocker, alpha plus beta blocker. But in beta, it is acting onto the beta 1 as well as beta 2 as well. Beta 1 as well as beta 2 as well. Exactly. Non-selective beta blocker. Exactly, guys. Fine. Now, next question. Long-acting beta adrenergic receptor agonist includes what? Albuterol, salmeterol, pyrbuterol, or orciprenaline. Yes. Easy. Easy. Non-selective blocker. Yep. Now, this one. Long-acting beta adrenergic receptor agonist includes what? Albuterol, salmeterol, pyrbuterol, or orciprenaline. Obviously, salmeterol is going to be the correct answer. If I talk about salmeterol, if I talk about formoterol, right? Formoterol, right? This is going to be the correct ones, right? SABA, LABA, long-acting beta-2 agonist, short-acting beta-2 agonist, SABA, right? All these things. Simple enough, right? Chalo. Salmeterol. Now, misoprostol, a prostaglandin analog, is useful as uterine relaxant, anti-ulcer, bronchodilator, or vasodilator. Chalo, easy enough. Yes, guys. Easy enough. Yes. What's going to be the correct one? That's your anti-ulcer, because misoprostol is a prostaglandin analog, just like PGE1 analog, right? PGE1 analog useful as anti-ulcer because it is going to form a mucous membrane. It is going to essential, that's a mucous membrane over here, right? Okay, fine. Anti-ulcer would be your correct answer. Simple. Yeah. Which of the following drug is most commonly used for prophylaxis of migraine? Prophylaxis: ergotamine, propanolol, methisargide, or sumatriptan. Chalo, easy. Yeah, prophylaxis of migraine. Prophylaxis means prevention of migraine, right? We can say in the treatment of migraine, we do have different drugs, but in the prophylaxis of migraine, we do have different drugs: ergotamine, propanolol, methisargide, or sumatriptan. Which is going to be the correct one over here? Which is going to be the correct one? That's your propanolol. Always remember prophylaxis pro and pro. Prophylaxis pro and propanolol. Propanolol will always going to give you the prophylaxis of migraine. Simple, easiest question, and plenty of time they have. In GPAD they have asked this question plenty of times. Okay, remember this one. Now, in order to perform the clinical trials on human being, the regulatory authorities issue which license? In order to perform the clinical trials on human being, the regulatory authorities issue which license? Good clinical practice, new drug application, investigational new drug, or none of these? Chalo. Easy enough. Yes, we have already discussed this one, right? We have already discussed this one. Yes. Good clinical practice, new drug application, investigational new drug, or none of the above? Hmm. New drug application. New drug application. It's C, bache. C. In order to start, in order to perform the clinical trials on human being, NDA is for marketing the drug, right? To launch the market into the drug. I mean, to launch the drug into the market, right? But to start the clinical trial, that would be simple. IND, investigational new drug application. Simple enough, right? Okay, chalo. Now, the term related to the detection, assessment, understanding, and prevention of adverse effects of drug is called to be as what? Pharmacoepidemiology, pharmacoeconomics, toxicology, or pharmacovigilance. Chalo, easy enough. Easy enough. NDA in phase 3, exactly. INDA is going to be the correct one. Yeah. Now this one, it would be very simple, right? We are talking about adverse effect. So adverse effect always. Pharmacovigilance would be your correct answer, D. Right? Now, which of the following is a type 2 allergic reaction? Type 2 allergic reaction. I told you a trick to remember. I told you a trick to remember if you remember into the class that I have given you a trick as well, right? What was the trick? What was the trick? Which of the following is a type 2 allergic reaction? Anaphylactic, cytolytic, arthus, or delayed hypersensitivity? Chalo, easy. Easy enough. Easy enough, guys. Chalo. Right? If you remember, I told you, yeah, I told you what? I told you A for 1, right? B for blood cytolytic reaction, blood cytolytic, B 2, right? C Arthus reaction, complex antigen antibody reaction, right? Complex antigen antibody reaction, which has an example of Arthus reaction. So C comes at 3rd position, D comes at 4th position. That's what I said. Anaphylactic, IgE, blood cytolytic. So cyto means 2, 2 means 2 antibodies, that is your IgG and IgM, right? Complex antigen antibody, the G over here, the G means IgG, right? And delayed, that's your T cells. Delayed, that's your T cells. Simple. This is the way that you can learn all these things. Clear? Now, so answer would be your simple cytolytic reaction, right? Now, which of the following is not an inodilator? Okay, inodilator. Inotropic in agent, but it causes a dilation. So, what's this? Levosimendan, enamrinone, milrinone, or tolvaptan? Not an inodilator. Yes, come on. Not an inodilator. Yes. Over here, that's your levosimendan. Okay, now quinidine acts as anti-arrhythmic drug by calcium channel blockade, sodium channel blockade, beta receptor blockade, or none of the above. If you remember, question produced difficality. Question: quinidine produce procainamide, D for diazopyramide, right? QPD, sodium channel blocker, class 1A, right? Class 1A. So that's for sodium channel blocker, quinidine. Simple. And if I talk about quinine, quinine, if I talk about quinine, guys, quinine, then it would be called to be as what? Quinine is going to be called to be as what? Yes, quinine is going to be given in which condition? Can you tell me? Can you tell me? Yeah, that's your anti-malarial in nature. Simple. Anti-malarial in nature. Also, we can say anti-amoebic or anti-anthelmintic. Anti-anthelmintic drug. Okay, that's also here, this one. All right. Sure. Let's go ahead now. Sodium channel blockade. Now, captopril is what? Captopril is calcium channel blocker, diuretics, ACE inhibitor, angiotensin antagonist. Right? Rupa levosimendan is an inodilator. Okay, Rupa, I have to see that one. Okay, I have to see that one, and then I'll let you know. Okay, now this one, captopril is an calcium channel blocker, diuretics, ACE inhibitor, or angiotensin antagonist. Captopril, all prils are what? All prils are what? That's your ACE inhibitor. What ACE enzyme do? ACE converts angiotensin 1 to angiotensin 2, right? है ना, angiotensin 1 to angiotensin 2. Simple. So ACE inhibitor is going to be the correct one. Now, muscle relaxant. Muscle relaxant of choice in renal and hepatic failure. Okay, muscle relaxant of choice in renal and hepatic failure. What's going to be the correct one? Pipacuronium, etracuronium, pancuronium, or doxacurium? What's going to be the correct one, guys, over here? Yes. Just an hint. Just an hint. That drug also undergoes Hoffman elimination. That drug also goes under Hoffman elimination. So what's going to be the correct one? Now tell me. That's your etracurium. Okay, etracurium, Hoffman elimination, right? Spatial rearrangement without any need of agency of any enzyme for elimination of the drug. That's your etracurium. Now, which of the opioid has maximum plasma protein capacity? Morphine, sufentanil, fentanyl, or pethidine? Yeah. Now tell me, which one of the opioids has maximum plasma protein binding capacity? Morphine, sufentanil, fentanyl, or pethidine? Yeah. Which one is correct? Easy enough. Easy enough. Morphine, sufentanil, fentanyl, or pethidine? Yes. That's your B, sufentanil. Okay, sufentanil is going to be the correct one. Now, anti-Parkinson agent which reversibly inhibits the enzyme COMT, ropinirole, entacapone, amantadine, or trihexyphenidyl. Chalo, easy, easy, easy enough. Yes, ropinirole, dopamine agonist, but that one is not COMT inhibitor. Entacapone, yes, entacapone as well as tolcapone. Entacapone and tolcapone are the correct answer. But there is one question. There is one question they used to ask sometimes, that which of the COMT inhibitor works peripherally as well as centrally? Then what you will say? Centrally and peripherally. Okay. Then what you will say, guys, over here? Is it entacapone or tolcapone? Tell me. Which one? Which one is it? Hmm? Which one is it? That's your tolcapone, exactly. Tolcapone acting both through centrally and peripherally. This is going to be the correct one. Okay, amantadine dopamine facilitator. Actually, it is a antiviral drug. Trihexyphenidyl. That's your central anticholinergic. Central anticholinergic to decrease down the acetylcholine levels. Okay, chalo. So entacapone is going to be the correct one. Now, indicate the competitive antagonism for benzodiazepine receptor. Chalo, very easy. Very easy. Benzodiazepine receptor. Now, flumazenil, picrotoxin, zolpidem, or temazepam. Right? Flumazenil, picrotoxin, zolpidem, or temazepam. Which is going to be the correct one? That's going to be obviously flumazenil, right? Flumazenil is going to be the correct answer. That's a competitive antagonism at benzodiazepine site, flumazenil, right? Now, which of the following carbonic anhydrase inhibitor that is effective as anticonvulsant drug? Uh, uh, Chalo. Answer is written over here. Paper has got leak. Paper has got leaked that which of the following carbonic anhydrase inhibitor that is effective as anticonvulsant drug? Acetazolamide, drug of choice for mountain sickness, if you remember. Drug of choice for mountain sickness. Okay, drug of choice for mountain sickness, and that's what I have taken when I was going for Ladakh. When I was going for Ladakh, this acetazolamide I have taken for 4 days consecutively. Okay, metazolamide, trihexyphenidyl, topiramate. Topiramate is going to be the correct one. Okay, topiramate is going to be the correct one. All right, chal. Next. Now, which is the drug of choice for maintenance therapy in uncomplicated bipolar disorder? Bipolar disorder. Kabhi the people is going to be very angry, or even once the people get very sad or depressed kind of a thing. That's what bipolar disorder, right? So over here, sodium valproate drug of choice for maintenance therapy, carbamazepine, lithium, or lamotrigine. What's going to be the correct one? Yes, easy enough. Yes, come on. That's your lithium enough. Hana? Lithium is going to be the correct one. Okay, now agranulocytosis is the commonly seen adverse effect of the following drug. Plenty of time this question has been asked. Plenty of times. Hana? And even by the other name also. Even by the other name also. Agranulocytosis. Agranulocytosis also demands, or you can say measure means decrease level of neutrophils. Decrease levels of neutrophils. Okay. This is the actual meaning of agranulocytosis. Agranulocytosis, decreased level of neutrophils. Right? I think in this year only in GPAT 2026, they have asked this question. They didn't written agranulocytosis. Instead of that one, they have written decreased level of neutrophils that is associated with chlorpromazine. No. Clozapine, yes. B is going to be the correct answer. Clozapine is going to be the correct answer. Chlorpromazine can also give you what? Chlorpromazine. That's for your partial seizures. If you remember, partial seizures, right? Partial seizure. Also, if I talk about Steven-Johnson syndrome, SJS, Steven-Johnson syndrome, lithium, you know it, haloperidol maximum, EPS, extrapyramidal syndrome, right? Extrapyramidal syndrome maximum in the context, haloperidol. Easy enough. Okay, chalo. Clozapine is going to be the correct one. Now, azapirones are useful in the management of what? Convulsions, anxiety, depression, or psychosis. Easy enough. Easy, guys. Yes, azapirones are useful in the management of what? Convulsions, anxiety, depression, or psychosis. Easy. Let's go. Come on. That's definitely going to be your anxiety. If you remember azapirones, buspirone. Buspirone is a drug which is your 5-HT1A partial agonist. Okay, agonist, and this is plenty of time has been useful in anxiety. B is going to be the correct one. Okay, azapirones. Now, all of the following drugs are atypical antipsychotics except olanzapine, clozapine, risperidone, or thioridazine. Easy, directly from the antipsychotics classification, guys. Chalo, directly from the antipsychotics classification. Yes. Which one is the correct answer? Yes, over here, that's your thioridazine, right? Thioridazine is your what? Thioxanthines. Now, effect of chlorpromazine include all except antiemetic effect, sedation, antipsychotic, hypertension. Effect, antiemetic, dopamine antagonist, antiemetic, simple. Sedation, it is having anticholinergic action. Anticholinergic action it do have? Yes or no? Yes or no? It do have? Antipsychotic, definitely. Hypertension or sedation? It between these two only, we do have one option that is right. Tell me, which is going to be the correct answer over here? effect of chlorpromazine. Yeah, which one? That's your hypertension. Yes, that chlorpromazine do have anticholinergic action, and that's why due to the anticholinergic action, it do have sedative activity. Okay, hypertension it doesn't have. Ranitidine is what? H2 antagonist, H1 antagonist, beta adrenergic antagonist, or alpha adrenergic antagonist. Ranitidine, furan ring, antagonist, but which? H2 antagonist, H1, obviously that is your H2 antagonist, simple. Okay, now the following classes of drugs are used in the gastroesophageal reflux disorder except, okay, GERD guys, GERD, gastroesophageal reflux disease, in which the lower, you can say the lower opening of the esophagus have reflex of the content that we have in stomach. Obviously, that is just because of the higher acidity. So, what drugs can we take at that time? Proton pump inhibitors, 5-HT3 blockers, sodium alginate, or antacids, except they have asked. Okay, except they have asked, guys. Tell me, which is going to be the correct answer over here? Yes. Proton pump inhibitors, PPI for gas. Okay, 5-HT3 blockers for vomiting. Ondansetron, 5-HT3 blockers. Ondansetron, we can give that one. Certainly we can, right? Sodium alginate, binder, antacids. Which one? 5-HT3 blocker we can't give. 5-HT3 blocker we can't give, guys. Okay, 5-HT3 blockers. Okay. In GERD, reflux disease we can't give, or we do not give generally. That's your ondansetron. Okay. Which of the following agents stimulate release of insulin from pancreatic beta cells? Stimulates the release of insulin from pancreatic beta cells. Glucose, incretins, dipeptidyl peptidase 4 inhibitor, or all of these? Obviously, simple. Glucose is the main reason. That's why insulin is going to be released. Simple. Incretins are also your glucagon-like peptide, GLP-1. GLP-1, right? Dipeptidyl peptidase, it's going to increase the GLP-1 level. So definitely insulin is going to be released, right? So all of the above, that's your correct answer. D is going to be the correct one. Okay. Now, अरे भाई सभी पेपर लीक करके बैठे हो. Now, which of the following is an adrenergic drug preferred for arresting labor? Whenever there is a uterus contractions takes place, right? So at that time, arresting means what? Stop. Arresting means stop. Hey, statue, right? Arresting the labor means relaxing the labor. So to relax the thing, ritodrine, isopranoline, salbutamol, terbutaline. Which is going to be the correct answer, guys? Yes. Which one is going to be the correct one? Arey bhai, ritodrine, salbutamol and terbutaline, isopranoline. These are also your uterine stimulant. I mean, relaxants. Is not ritodrine. I would say phosphodiesterase 5, right? Phosphodiesterase, right? Hana? Ritodrine is your phosphodiesterase 4 inhibitor. Phosphodiesterase? I don't know exactly. I'm not able to remember all these things. Chalo, fine. Let it be now. Let it be for now. Now, find the correct option for match the oral hypoglycemic drug with its category. Chalo, tell me. Which one? Tolbutamide. Okay. Tolbutamide, dapagliflozin, bromocriptine, acarbose, and metformin. All right. So tolbutamide, sulfonylurea. Yes, exactly. Dapagliflozin. No, that's a SGLT2 inhibitor. SGLT2 inhibitor. Bromocriptine, dopamine D2 agonist. Right. Acarbose, alpha glucosidase. Right. Metformin, biguanide. Right. So Q is wrong. Okay. So P and Q correct? No. P and ST all correct, but Q is incorrect. Yeah, B is going to be the correct answer, right? B is going to be the correct answer. All right. Chalo, fine. Now, Let's go ahead now. Now, Maladi is famous. Maladi is famous drug as contraceptive. Find out the drug which is present in the Maladi drug. What's that? Norgestrol 0.3 mg, ethanylestrol 30 micrograms, levonorgestrol 0.25 mg, ethanylestradiol 50 micrograms, desogestrel 0.15 mg plus ethanylestradiol 50 micrograms, levonorgestrol 0.25 mg plus ethanylestradiol 50 micrograms. Chalo, easy enough. Easy enough, easy. Chalo. Which one is correct? Which one is correct over here? Yes. That's your A, norgestrol 0.3 mg plus ethanylestradiol 30 micrograms. Now, antidote for overdose of fibrinolytic therapy. Okay, fibrinolytic therapy ke against we do have what? Antifibrinolytics, simple. That's your tranexamic acid or epsilon aminocaproic acid. So we do have epsilon aminocaproic acid. Yes, exactly. A is going to be the correct answer. Heparin, if I talk about heparin, is your parenteral anticoagulant. Protamine sulfate. Protamine sulfate is an antidote for heparin. Warfarin is your oral anticoagulant, right? For oral anticoagulant, vitamin K is your correct antidote. Epsilon aminocaproic acid, or we can say tranexamic acid. Okay, tranexamic acid. Okay, tranexamic acid is going to be the correct answer. A. Fine. Now, oral active drug directly against factor 10A. Okay, directly against factor 10A. Argatroban, rivaroxaban, lepyridine, or dabigatran. Chalo, tell me. Which one is the correct one? Simple, 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 simple. Yes, come on. That's your rivaroxaban. In the name itself, you can say 10A. Here you can see factor 10A, rivaroxaban. Rivaroxaban XA, 10A ban. Okay. B is going to be the correct one. Yes, exactly. Very nice. Okay. Now, a parenteral anticoagulant drug is what? Parenteral anticoagulant. Penicillin, phenandione, warfarin, or heparin. That's all we have discussed before as well, right? Before as well. Penicillin, it's not an anticoagulant, obviously, that's an antibiotic drug. Phenandione. Phenandione is what? Phenandione is your vitamin K1, right? Phenandione. Warfarin, yeah, warfarin is your oral anticoagulant, whereas if I talk about heparin, so parenteral anticoagulant. B is going to be the correct one. The storage form of iron in the human blood is what? Ferritin, ferrous fumarate, home, home, or globulin. Chalo, easy enough. Ferritin, ferrous fumarate, home, or globulin. Yes, easy, 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 easy. That's your ferritin, right? A is going to be the correct one. Which among the following is an oral acting antithrombin drug? Oral acting antithrombin drug. Oral acting. Hirudin, lepidolin, bivalirudin. These are all parenteral. These are all parenteral indirect acting drugs. Dabigatran, direct oral acting drug. Direct oral acting drug. That's your dabigatran. Right? D is going to be the correct answer. D. Simple. D is going to be the correct answer. Now, the first vaccine was developed by Louis Pasteur, Edward Jenner, Karl Landsteiner, Joseph Mister. In KBC, 2020, there was a prize money of 6,40,000 rupees. 6,40,000 rupees. The same question they have asked in Kaun Banega Crorepati. 6,40,000. Kaun Banega Crorepati? They have asked this question. The first vaccine was developed by Louis Pasteur, Edward Jenner, Karl Landsteiner, Joseph Mister. Chalo bhai. Easy. Easy. B. Everyone has got 6.4. है ना? Edward Jenner. That's for smallpox vaccines. Okay. Now, which vaccine is given soon after birth? Polio, varicella, hepatitis C, measles, mumps, or rubella MMR? Chalo, easy. Yeah. Which vaccine is given soon after birth? Yeah. Polio, varicella, hepatitis C, measles, mumps, and rubella MMR. Which is going to be the correct one over here? That's your polio. Yes. Now, oral polio vaccine, if you remember, oral polio vaccine, right? Now, which type of bioassay generally provides greater accuracy than a matching assay? Single point assay, multi-point assay, physical assay, or chemical assay? Single point assay, multi-point assay, physical assay, or chemical assay? Yes, easy, very, very easy question. Very easy question, guys. Yes, single point assay, multiple point assay. More accurate. More accurate. How many of you have done this one? How many of you have done this essay in your pharmacology practicals when you were in third year? That's your multiple point essay. Okay. Now, oxytocin bioassay is commonly performed using what? Isolated uterus, isolated heart, isolated ileum, or isolated trachea? Chalo. Easy. Easy. Yes. Oxytocin bioassay is commonly performed in isolated uterus, isolated heart, isolated ileum, or isolated trachea. Yep. Who will give me the correct answer? Who will give me the correct answer? That's your isolated uterus. Fine. Nice. Chalo. Alright, guys. I think this is the end from my side and even from the whole this panel of me, Pratik sir, Rajeshri ma'am, and Girja sir. Okay, so this is the end, and I hope that I have made you clear around all the concepts that we have discussed in this session of mine. And just give it a like, guys, and just do a comment for all the faculties of GDC English if you like their dedication and you love all those things, right? Whatever you want to say, just do come after 5 minutes. Do come after 5 minutes when this live streaming is going to be end, and then do share a message, whatever you want to. Okay, so this is it from my side. I hope I have given you a lot of concepts with correct answers. There are a few, but see. Okay, so still, thank you so much, and wishing you all the very best for the rest of the preparation. Thank you so much, guys. Good night. Bye bye.